Hey everybody, today we are debating human evolution and we are starting right now. Ladies and gentlemen, thrilled to have you here for another epic debate. I'm your host, James Kunz, and welcome to Modern Day Debate. This is going to be a fun one, folks. So we have two seasoned debaters with us, two very clever and quick on their feet type debaters. This is going to be a lot of fun, folks. So want to let you know, if it's your first time here, consider hitting that subscribe button, as we've got a lot more debates coming up. We are very excited. In fact, I got to show you just a few of these because they're going to be epic. So first, Aron Ra will be facing off with Dr. McElroy. That's going to be a big one, and that's going to be in person. So we're actually going to be streaming that, live streaming it from Dallas on our little, you could say, mini debate tour. So that's going to be a big one. Then, also want to mention, Nathan Thompson and Team Skeptic will finally cross swords. So they are going to be debating a flat earth debate. That's going to be a lot of fun. That will also be in Dallas. Whew. Hopefully, things don't get too crazy. It might, who knows? I don't know. It's going to be, we hope it's not a fist fight. We'll be all right. We're going to make it, folks. Next up, I want to mention, if you haven't seen this, where have you been? Have you been living in a cave under a rock? Michael Jones from Inspiring Philosophy and Matt Dillahunty will be facing off during our Texas debate tour as well. So, folks, I am pumped for that. It's going to be a lot of fun. That will be streamed live and in person as well. So, want to let you know, for today's epic debate, both of our speakers have their links in the description. So if you're listening and you're like, I like that, I want more, well, you can get more just by, it's like that little description box down there. You can find more right there. So, with that, dear goodness, I just realized, remind me, the format, if I remember. Oh, that's right, I do remember now, thanks. It's a 10-minute flexible opening. So that means, we'll give them, a little leniency, but, once it gets up to 12, I will be so happy to cut Standing for Truth's mic. I mean, I will. It's going to feel terrific. So, I told him, I said, don't go over 12. Don't even think about it. So after that, we're going to jump right into open discussion, which I think most people, it's a lot of fun there, followed by Q&A. So if you have a question, fire it into the live chat. I will pull those questions out of the live chat, and super chats go to the top of the list, and also allow you to make a comment during the question and answer if you'd like to do that. So with that, very excited, folks. We're going to jump right into this. And so thank you very much, Erica and Standing for Truth, for being here, first of all.
Well, do we get to comment on that? I'm excited to be here. I'm ready to have a good time on this potential eve of World War III that Twitter's been going on and on about. So there's nowhere I'd rather be than here.
Glad you are here.
And that goes for me as well, James. I'm really looking forward to it. Love this channel. Everybody, please like and subscribe. Hopefully, Erica and I can help make people's night a little bit better after the yes, like Erica said, the World War III scare. Hopefully not.
Yes, we hope everything is safe with that. And so it's been sort of a wild day, and we are glad to be here with you together. It kind of gives you the big picture. We've talked about this before. It's like we debate. Sometimes we even get a little snippy with each other. But at the end of the day, we agree on the big things, like hopefully making the world a better place, stuff like that. So with that, very excited to get the ball rolling. And if I remember right, Standing for Truth, are you getting the ball rolling, or?
Ooh, I think it might be me.
Erica, sorry about that, you guys.
Yes.
Long day.
Yes.
Fair enough.
No, please, don't worry about it. Yeah, I think it's because I'm the affirmative, but I'm not positive.
Deal. And I'm going to set the timer for, like I said, 10 minutes. I'll just let you know once you get up to 12 if you get there. And Erica, the floor is yours. Thanks for being here.
Okay, cool. So my name is Erica. I go by Gutsick, Gibbon on YouTube, do a lot of videos on anthropology and bioanthropology and primates because, God, I love primates. I got a BSA in animal science, picked up a minor in biology and one in anthropology along the way, and I'm currently getting my master's degree, a master's of research in primatology, hence why I love the primates so much. So, human evolution, an undeniably wonderful journey. Now, from my standpoint, there is indeed so much support for human evolution across numerous fields that cramming it into a 10-ish minute opening statement is going to be kind of tough, but I am going to try to do it. Now, as I mentioned last time in my conversation with Kent Hovind, even Carlos Linnaeus, who was a creationist, admitted that humans are indeed primates because he recognized that we were eukaryotes, animals, chordates, vertebrates, mammals, eutherians, and so on and so forth down the line. And he also realized that similar to how this rhesus macaque here has its nostrils opening downward and how it has the dental formula of 2123 and shares 93% of our genetic similarity, we are both catarrhines. So the same reason that you would classify this rhesus macaque as a catarrhine, you would use to say, oh, well, I guess humans fit into that category as well. And I happen to find that very inspiring and cool. So we don't have to just use morphology, though. We can also have a conversation about paleontology, which happens to be a favorite of mine, because evolution can indeed make a prediction. If evolutionary theory is accurate and humans did indeed evolve from an ape-like ancestor, which we would currently in the field consider Sahelanthropus chadensis, then we can say that if we share a common ancestor with chimpanzees, we should see the emergence of human morphology through geologic time. And paleontology displays this gradient from our ape-like common ancestor with chimpanzees, Sahelanthropus chadensis, to modern humans. So essentially what we're saying here is if this is indeed the case, we should see slowly through time the appearance of novel human traits. And with this comes the macroevolutionary concept of speciation. So while B may not be able to interbreed with E, B may have been able to interbreed with C. And through this concept of small, novel microevolutionary changes, if you like, we do indeed see evolution occur. But of course, that is only the case if we are seeing this massive change occur through geologic time. And that is indeed the question. I just threw this one. Forgot I had this one in here. I love Australopithecus africanus almost as much as I love Afarensis, which would be Lucy. But as I asked Kent in the last debate, if you were to pick one of these, he likes to do they have a 5-year-old come up and tell us which one is the difference in order to differentiate biblical kinds. If you were to do that here, you'd be kicking the chimpanzee out and you'd be keeping Australopithecus africanus right up here in the mix. And of course, it has a myriad of lovely mosaic traits from the semi-parabolic palate, that is to say the shape of the upper palate, ventral frame and magnum that allowed it to stand indeed bipedally, which I do hope we have that conversation today because I think it's quite a good one. Sort of some retained neoteny that's continuing, and a pelvis that is, of course, looking very human-like. But like I said, can we prove that all these organisms, according to the evolutionary model, did indeed live at slightly staggered different times over the course of millions of years and not all at the same time and then perished in a global flood? Yes, we can indeed do so, thanks to the radioactive decay law. Now, the radioactive decay law is essentially this process of spontaneous transformation, essentially decay, rather, of a parent isotope into daughter isotopes. And when we look at these minerals that we find fossils in, we can get what's called an absolute date by looking at this ratio. Now, the radiometric decay law, radioactive decay law, is indeed a firm law in physics. It additionally covers the nature of decay constants and half-lives and indicates that to our current knowledge, decay cannot change in any meaningful ways in nature. Because frequently, what creationists tend to do is they say, oh, well, maybe it worked differently in the past, or maybe the flood accelerated this radioactive decay and gave us these dates that seem to say millions of years. But there's a problem with that. And that problem is when things decay, they release a ton of heat. Now, we're going to talk about that in just a second. But the RAPE group, which is a young Earth creationism group, they were discontinued back in 2005. They spent a ton of time trying to prove that you could indeed accelerate this without problems, but they couldn't. And this is a quote essentially from that report. They admitted that a young Earth position cannot be reconciled with the scientific data without assuming exotic solutions will be discovered in the future. No known thermodynamic process could account for the required rate of heat removal, nor is there any known way to protect organisms from the radiation damage. And interestingly enough, a Tufts geologist went ahead and decided, all right, let's see what happens if we try to cram all the heat that results from accelerated decay from a 4.8 billion year old Earth to a 6,000-year-old one. And it's not a good situation, folks. He essentially found that at the time of Adam and Eve, if you're cramming all of that heat release into a 6,000-year time scale, the surface of the Earth is going to be 70,000 degrees Celsius for every square kilometer. And at the time of Jesus' birth, assuming a generous geothermal gradient, we're going to see around 400 degrees Celsius for every square kilometer. There's a link at the bottom there, and I could give to James if you'd like. But essentially, As far as we know, radiometric dating works because there's no reason that it shouldn't. There's no empirical question that has essentially been raised to say, well, what if it did work differently? It's rather firm. But of course, genetics are some of the more interesting and novel supports for human evolution that have popped up recently, because when you align our genomes side by side with the members of genus Pan, that would be the chimpanzees and the bonobos, you get a 98.8% similarity when you're coding for, or when you're comparing coding base pairs, which is fascinating and very cool, because when you compare their genome to the rest of the apes, you find that they are actually our closest living relatives just as much as we are theirs. They share more in common genetically with us than they do with a lowland gorilla, which is really cool. Now, I said this to Kent as well, but essentially that genomic comparison I just mentioned is just a souped up version of a paternity test. So the key thing to take home here is that if relatedness is determined by genetics in humans, why isn't it the same across species? But we can also look to the chromosome 2 fusion. So human chromosome 2 is a fusion of 2 chromosomes found in the other great apes. How do we know this? Well, this is a little picture of a chromosome, and it's tipped with telomeres. Now, the cool thing about that is telomeres have a very specific identifying sequence, which is TTAGGG, and its reverse, which would be AATCCC. They also have centromeres in the middle. So essentially, if you are accepting of evolution, you're going to make a prediction that human chromosome 2 is a fusion of these 2 hominoid chromosomes, 2A and 2B, sometimes 12 and 13 is what they're referred to as. So we should find telomeres in the middle of human chromosome 2, because if they're fusing, right, they should fit together like that, and you should find endpoints in the center of our chromosome, with the upper portions of that chromosome being nearly identical to those separate chromosomes in the apes. So is that what we find? Yes, it is. It's very cool. So the banding in human chromosome 2 matches almost precisely to the banding that chimpanzee chromosomes 12 and 13, 2A and 2B, like I mentioned earlier. And then you have this inactive centromere in the center. Interestingly enough, and I do hope we get to talk about this as well, but this centromere is known to be a centromere, even if it may have a functioning gene on top of it, because it does indeed contain that TTA-GGG sequence that I just mentioned. So this is a prediction that evolutionists made that did indeed come true back in 1991. It's the title of the paper. It's a good paper, a little lengthy. So we could also talk about the gulo breakage. So the gulo is essentially a broken gene that in most mammals allows them to synthesize ascorbic acid so that they can make vitamin C and they don't have to eat it, whereas we have to eat our vitamin C. Essentially, this would have broken around 61 million years ago with the omomyids and the adapids, which are little almost tarsier and shrew and lemur-like guys. This means that, like I said, we can't synthesize our own vitamin C. But the interesting thing is this breakage is in the same location in every single primate that is a haplorhine and onward. And furthermore, while gulo breaks also occur in other mammals, it's only in the primates that we get the CAMP gene expression by vitamin D. So what creationists need to invoke here is a separate occurrence of this breakage in every single primate lineage, as well as a separate CAMP alteration. That's a hard sell for me. We could also talk about the trouble with tools. So I didn't bring this up last time, but I think it's quite an interesting one. So essentially, there's trillions of tools strewn all across the African continent. They're typically these napped tools that we see, like Oldowan and Oldowan style tools that you get by taking 2 rocks and kind of chipping them together in like the classic caveman sequence. And there's no natural way that these tools can form. You can look at a cool picture of them. You're not going to get this by rolling a rock down the hill. But we have 15 to 150 trillion with the T of these guys laying all across the African continent, based off of an estimation of 250,000 stone tools found in approximately, per square kilometer in Libya, which is a whole lot of tools. So yes, it's a bit extrapolated, but let's see, 250,000, that's quite a few in and of itself. So the question is, how do creationists deal with all these tools? Because under an evolutionary lens, right, we've got several million years for organisms like Kenyanthropus platyops or Homo habilis to make all these tools and leave them all around as they migrate across the continent. So it's basically a numbers game, because young Earth creationists kind of hold, generally speaking, at least Answers in Genesis does, where I got this lovely timeline, that there's a Stone Age that occurred approximately 500 years maximum. Yeah. And this would be essentially when everybody left the Tower of Babel. So you've got 500 years, you've got 25 years per generation with a replacement rate for the population, 10,000 artifacts produced by each person in their lifetime. and an average population size of about 100,000. Can you get all these tools with those population rates given by answers in Genesis? And the answer is no, you can't. It's very problematic. So young Earth creationists essentially have to have a small band of people leave the Tower of Babel in 2250 BCE, migrate to Africa, forget all of the technology they once had in Mesopotamia, learn how to fashion rocks into tools, which is a technology in and of itself, and extract 40 million pyramids worth of rocks from all over Africa, fashion them into 15 trillion, and that is the lower end of stone tools before placing them all across Africa in approximately 150 to 500 years. To steal from Kent here, that's imagination stuff. It's not going to happen. So essentially, to prove human evolution wrong in this context here, you're going to have to invalidate genetics by relatedness and genetic similarities in human genus Pan across their genomes, the human chromosome 2, and the Gulo break. We're also going to have to produce a mechanism for accelerated decay that solves the heat problem, which I think is going to be somewhat difficult. We're also going to have to introduce morphologic means of separating the humans from the rest of the apes. And we're going to have to produce a mechanism for those tools to form naturally, or a proper population growth that is going to account for all of them in such a small time frame. So I'm going to cede the rest of my time because I'd like to get to the discussion.
Thank you very much, Erica. Next, we will go over to Standing for Truth for his opening statement. And Standing for Truth, I have got the clock set, and the floor is yours. Thanks for being here. Bet you have yourself on mute. Whoa, wait a minute. I think. Oh, no, okay, full screen. Okay.
There it is.
I thought I lost you. Okay.
Let me move this to the side. Okay, let me start my timer as well, just so I know. Let's see here. I got it set max 12 minutes. Okay, let's go. Okay, thanks so much for your opening there, Erica. I'm going to try and cover as much as I can. I've got my opening here. I'll address a few things that she talked about earlier. For the decay rate, according to catastrophic plate tectonics, if rocks were exposed to heat and pressure, this could speed it up. Heat from below the crust coming up to the ocean, then to continents, of course. So volcanic activity hitting rocks worldwide. She talked about human chromosome 2 fusion. Well, that purported fusion site's actually a functional DNA element in a human gene, so that's been overturned. When it comes to the human-ape similarity, they say it's about 99%, maybe 98. New incoming data suggests that it's more like 88% similar. That's 400 million DNA differences that separate apes and humans. Also, the chimp genome is 12% larger than humans, so that helps separate us from chimps. They ignore things like copy number variations, and of course, there's huge gaps on human and chimp DNA. So let's get into it here. Debate human evolution. Looking forward to this one. Let me see. Okay, can you see my slides?
You can see them.
So she talked a lot about nested hierarchies as well. I'll get into that. There exists, though, an unstoppable crisis for those that believe apes, humans, and banana plants are related through common ancestry. Now, this crisis is that of genomic degeneration. The evidence is absolutely overwhelming that mutations are accumulating. I'd like to ask Erica to provide us with a type of selection or mechanism that can rid the population of so many deleterious mutations that are mostly unselectable and invisible to selection. So unfortunately for human evolution, which is the topic of this debate, both ape and human genomes are degenerating due to this mutation accumulation. This evidence is extremely strong. This strongly indicates that neither ape nor man could have evolved. They both must degenerate. In fact, many of the hominin fossils here that Erica uses to prove ape to man evolution clearly display anomalous pathologies that indicate that in the past, several isolated and inbred human populations have experienced reductive evolution. This goes for Homo neanderthal, Homo erectus, Homo naledi, the hobbits, of course. There is no progression. This type of reductive evolution has resulted in genetic pathologies, reduced body size, reduced brain size, and as a consequence, tribal extinction. This is expected given a biblical-based model of post-Babel conditions and post-Babel dispersion. Genetic degeneration is a very real phenomenon occurring in living populations. If what we observe today in the present is reverse evolution and not forward evolution, the same thing would have happened in the past. And it is very well documented that what we truly observe in the hominin fossil record is genetic degeneration. For example, numerous of the bones of the alleged Homo species like Hobbit, Erectus, Naledi, Neanderthal, the ones that she used here in her opening, appear to demonstrate testimony of severe inbreeding and genetic decline. Therefore, many of the troubling human fossils that Erica used in her opening presentation are much better understood to be devolution rather than evolution. The so-called primitive features that Erica here points to as a progression from ape to man is actually genetic degeneration. They weren't pre-human tribes. More accurately, they were human tribes that were persistently inbred and were in genetic decline. We can see here a paper actually that the claimed primitive humans are inbred subpopulations. This was a paper by Eric Trinkos. You can see it here. He's a paleo expert, and he noted that a total of 75 documented developmental abnormalities from 66 individuals spanning the Pleistocene. The author also explains that the remarkably high frequency of skeletal pathologies could not possibly be due to chance. This is exactly what we'd expect based on a post-Babel dispersion of isolation and subsequent inbreeding. The author suggests that these populations were almost certainly undergoing genetic degeneration due to inbreeding, as is expected in any small and isolated population. So these human variants, they all lived in isolation, and given the situations associated with each variant, the specific settings would lead to these founder effects, isolation and inbreeding. And what's funny is the author obviously doesn't conclude the biblical base model, but these findings and more strongly support the biblical base model of degeneration of small subpopulations, which is exactly what our model talks about. We got here just recently, I believe it was in 2017. This negates all her dating methods because we've got fossil footprints, anatomically modern human-looking footprints that debunks human evolution in the same layers or lower than the Australopithecines. So anatomically modern human-looking footprints have been discovered here in Crete. I believe it's in Greece. They date approximately to 5.7 million years old. So these findings here are clear evidence that Lucy, Ardipithecus, Australopithecus sediba, Homo habilis, etc., they're not our evolutionary ancestors, as Erica promotes here. And the reason is because these footprints indicate that creatures with human feet lived much earlier than these so-called human ancestors. So Erica's going to have to answer this question if she wants to look to rocks. It's genetics, genes, traits. These are what are inherited sperm and egg, not a rock, not geology, not geography, but genetics. So she's going to have to explain these anomalies to her evolutionary theory. I think this article here on Evolution News puts it perfectly. They say, when the oldest known evidence for hominin feet predates the alleged African ancestors such as Artie and Lucy, but already shows relatively modern human footprints, what is more congruent with this new evidence when looked at without bias? A gradual Darwinian evolution, or rather a saltational origin that requires intelligent design? Just a few points here. Evidence from paleontology, archaeology, and modern genetics all irrefutably confirm that Neanderthals were in every way fully human. Erectus, for example, was able to sail, which clearly speaks of human intelligence. Homo floresiensis, which Erica helped me say in the pre-show, thank you so much, was also very clearly human, as indicated by their impressive cultural inventory, ability to sail, endocast scans that reveal a modern human brain, an anatomy that is modern human looking. Reduced brain sizes, for example, can be beneficial in certain conditions, such as starvation. This explains the smaller brain sizes that we do see in some of the hominins. This is because brains are energetically costly. This is all evolution in reverse, and these pathologies and variants can occur quickly in isolated populations. This is all expected and predicted in the biblical model, post-Babel and post-flood. Nested hierarchy, she spent some time on. before we even had DNA sequencing technology, it's quite obvious at the physiological level, humans and the great apes had a lot in common in terms of appearance, basic body structure. Both mammals have binocular vision, etc. There's some major differences, of course, though. Just look at the feet of the great apes and the shape of their feet. They have feet that look like hands. They use these for grasping purposes, of course, swinging from trees, etc. Now, humans have feet, on the other hand, that are built for upright walking. We are similar in these areas, and therefore we should have similarities genetically as well. This should not surprise anybody, and this is in no way, shape, or form proof that humans, apes, and pine trees are related. Now, of course, evolutionists like to look at these similarities and imagine that all life forms are related, but this is not empirical. This is actually in the realm of fairy tale. The bigger question we are trying to answer is, why are humans and great apes so similar? It doesn't matter whether we look at the anatomy, the physiology, the genetics. They all tell us that we are a lot more closer to each other than pine trees and dogs. Now, evolutionists will say the similarity genetically is the high 90s, like Erica was saying. It's just false. It's more like 88%. But regardless, we are pretty close. What does this tell us about our origins? The evolutionist seems to think to themselves that if we really are made in the image of God, shouldn't we as humans be distinct from the rest of biology? Well, maybe in a way, yes, but in another way, no. The Bible doesn't imply in any way that we have biological life and then human, which has absolutely no similarity to any other species out there. There was no reason for us to believe that based on a biblical model, there should be no type of hierarchy in the biological world. There's no reason to believe that we shouldn't be more similar to one species, for example, the apes, than to another species, for example, a wolf. Naturally, we as humans have designed things with groups within groups patterns. If you were to look at the types of vehicles or modes of transportation humans have created, they naturally form groups within groups patterns. A sedan has a tremendous amount of similarities with other sedans. Now, a semi-tractor trailer is obviously much different than a sedan or a car, but these two modes of transportation are similar enough to each other that it sets them apart from airplanes. This is the groups within groups patterns that emerges when we look at all the modes of vehicles or transportation that humans have designed. This is an empirical observation, and this is something we naturally do. It appears that God created kinds in the beginning that are different from us as humans, as well as kinds of creatures that are more similar to us. This is a reason why Aron Ra's phylogeny challenge is fallacious. We too predict nested hierarchies, morphology, anatomically, and genetically. Evolutionists miss the bigger picture. Yes, we can find hierarchies in all of biological life, including humans and apes. But apes don't build airplanes. Apes don't write books. Apes don't compose symphonies. Chimpanzees and other animals don't do any of these things. Mankind has built the space shuttle and has accomplished wonders. So yes, there are groups within groups patterns in all of life, but this is simply a hallmark of design. The fact that there exist these natural and nested hierarchies is exactly what we'd expect if God created the world. We can see here, even if we got into transitional forms, we create things that blend the features of two different types of vehicles, amphibious assault vehicles, for example. We can see here that all the evidence regarding mitochondrial DNA, Y chromosome, the low genetic diversity that we see all points to Adam and Eve. Evolutionists need to ask themselves, if they have an open mind and they were to look for evidence for Adam and Eve, what would they expect to find? Because all human beings are profoundly similar genetically, and this points directly to creation. I'd like to talk to Erica here about the evolutionary out of Africa bottleneck and how that may be feasible.
1 minute.
I've got 1 minute here. Therefore, going into the discussion, a few questions that I would like to ask her are: the out of Africa scenario, due to the resulting inbreeding of a population bottleneck of 10,000, how is this even remotely feasible? I'd like to ask her about the Y chromosome of apes and man and how they're so dissimilar, less than 70%. The Y chromosome should be the most similar. So I want to ask her that question, as well as genetic degeneration. These near neutral deleterious mutations are accumulating. What type of selection can stop this genomic degeneration? I've got a lot more here, but for the sake of time, we'll save some of that for the discussion. Thanks so much.
You got it. Thank you very much, Standing for Truth. We will now shift back to the dialogue boxes and let her rip. So, thanks for your questions so far, folks. A lot of great questions. Excited to ask those. And now we'll go into open discussion. So the floor is yours.
Okay, well, there's definitely a lot to talk about. First, I would like to quickly say they have actually tried time and time again to speed up or to accelerate decay with heat and pressure. And the most that they were able to do was 1.5%, which I believe it was. I think it was Woodmarap, who's a young Earth creationist, commented on it and said, "Yeah, that's nowhere near good enough. We're going to have to, invoke some exotic solutions here." So, I mean, it's easy to say, yeah, we could just have a lot of heat and a lot of pressure, but that's never been demonstrated in the slightest to be good enough to get it anywhere close to what you need, especially because that 1.5% increase of decay wasn't even in a geologically relevant element. But more specifically, I would like
Well,
to talk
if I could just respond to that real quick.
Yeah, sorry, go
Um,
ahead.
No problem, no problem. I do think that the entire, radioisotope dating, the methods, for example, they do have unprovable and unknowable assumptions, right? For example, are the initial conditions of the rocks known? Clearly they aren't. So geologists, they weren't there when the rocks form. So at the end of the day, they don't know, right? For example, how do they know how many uranium atoms, uh, if they had no lead atoms, for example? But I think it comes down to the fact that we do find now these anatomically modern looking human footprints in layers, either with the Australopithecines or lower. Therefore, how can there be modern human looking footprints in layers with our so-called Australopithecine ancestors? So
Yes. So I would love to talk about that Crete footprint because I have looked quite extensively into this. I had this question asked of me about a year and a half ago on the Debate Evolution subreddit. But to quickly kind of take a step back and make kind of a comment on that, on the radiometric decay thing. Yeah, we kind of do know the starting ratio because we do have rocks that do indeed form today. I see no reason why, since we've literally never observed a rock forming that didn't have a complete, parent favored ratio of isotopes, why we should expect that back then it was different, especially because, and of course, I'm sure we can have a conversation about mutation rates. There are reasons why we do say, well, some rates are constant and some are not, but there is, of the hundreds upon hundreds of thousands of times that we've radiometrically dated things and observed new rocks form, we've never been given any reason to suggest that ratio wouldn't be in favor of the parent, and the physics inform that. Um, but again, I probably shouldn't have included that stuff in because this is a conversation about human evolution. It's kind of my bad. I guess I was a little
well, no
baby.
problem, no problem. I definitely don't like to leave anything hanging because you did
Yeah,
bring up
yeah,
the
sure,
heat
sure.
again. I just want to say, for argument's sake, I mean, I do stand behind the fact that it's genetics, it's traits, it's our genes that are inherited sperm and egg, not a fossil, not a rock. So, I think we should definitely look to genetics, morphology, anatomy in
Sure.
regards to human evolution. But because of the heat, I do want to say, based on our model of catastrophic plate tectonics, right? Subduction, friction, for example, with the continent, will make heat and make the rock more plastic. So indeed, this subduction can happen rapidly. This is actually called runaway subduction, or speed up the subduction, which can explain the necessary speeding up of the decay rate. But in regards to the hominins, which I do want to focus on, I'll let you comment on the footprints in Crete as well. But as far as I know, the dating methods commonly used to date the hominins, they do yield extremely inconsistent ages. Therefore, the paleoanthropologists seem to only use the dates that fit their story, right? So the dates that conflict with the proposed ape to man timeline are routinely rejected. Like, I'm guessing that these anatomically modern looking human footprints, the dates are probably going to have to be changed now, right? Because they've done it so many times before. These unacceptable dates are routinely assumed to be corrupted by sample contamination or whatever reason that they have, even if they have no evidence for said contamination. So it just seems like selective use of data. And you mentioned the genetics and the mutation rates. Well, I mean, we have a molecular clock regarding the Y
Sure.
chromosome and the mitochondrial DNA, which actually brings us back to 6,000 years. I mean, if you want to talk about dating, we can look at traits, genes, and genetics. That
Yes,
gives us a date of 6,000 years. Go ahead, Erica.
well, I'm totally with you, but that, I guess I kind of opened Pandora's box there because that was a lot of points. So I would very quickly, I'm going to make a comment on Crete, on that Crete footprint. Have you seen a picture of it? Because I have. Did you look up a picture of it?
Yes.
Yeah, it's not very footprint-esque, especially considering what trackways tend to produce, especially given the sediment that it's kind of placed in. It's very unlikely, at least amongst the professionals that I have spoken with, that is actually a footprint at all, specifically because it's a single print. It's not, there's only one print that's being proposed. It's not a trackway like the Laetoli footprints where you've got, dozens of footprints with where you can actually take a plaster cast and get the balls of the feet and the toes and the heel, and you can actually see how the individuals that made those tracks bear their weight. You can't do that with the one in Crete. And
Right.
I tend to think, if you go out and you pour a bunch of water in the mud and then you walk through it, you're not going to get one footprint. So I actually, everything I have seen on the Crete print suggests that it is not a print at all. And at least, and moreover too, one print, let's say you could prove that it was a print of something, right? That wouldn't overturn the entire timeline that we have now, particularly because we know for a fact that Europe used to have a booming population of primates. It would indeed be entirely possible that you would have apes living in the Oligocene and the Eocene.
Isn't it true that these footprints were found outside of Africa? And are you saying there was only one set of footprints that were found? And because from the article that I had posted in the opening presentation, the headline does say ancient footprints in Crete challenges theory of evolution, but what actually made them? I've looked at the source material as well, and I didn't find any data suggesting that they weren't actually footprints. I saw some paleoanthropologists or some scientists in the field claiming that maybe it was some type of extinct gorilla that had a footprint very similar to modern humans. But I didn't see anything about what you're claiming. So I'll definitely look into that. But if they are footprints, especially if they're outside of Africa, I think they're near Greece.
Yeah, they are.
That could challenge the theory.
It would add a lot of nuance to it if they were indeed footprints. But again, I would heavily suggest looking into it because I read the paper and then I looked for commentary on the paper, and then I also asked a past professor of mine kind of what his thoughts were. And everything that I've read is suggesting, one, it's very likely not not prints, because all I could find was, as far as pictures go, was a single kind of imprintation in the sediment that does, first of all, it doesn't particularly look like it would be in the shape of what you're kind of proposing as an anatomically modern human print anyways.
Would you
it's much
are you saying that it looks totally
hold on. Oh, forgive me.
Sorry.
Just to be sure that because there's a lot of info going back and forth, I want to be sure that she gets plenty of time to respond.
Of course, go ahead, Erica, take your time.
Yeah,
I just
sure.
want to say one last thing about the footprints when you're done.
Yeah. I mean, my point is that it's not, even if it were a footprint, which there's very, very few people standing up and saying that it is. And even if it were a footprint, there's even fewer. I don't know of anybody saying this is a hominid, a hominid. It's the wrong shape if you're trying to propose that this is anatomically modern. That's all I wanted to say about that. I don't find it very compelling.
If you're done, I definitely want to see. I'd want to see the paper or the source that says that they're not really footprints, because I've looked into it thoroughly and I never seen anything of that. So I'll definitely reserve judgment till I see that paper. But you did mention Mary Leakey, which about 25 years ago discovered some footprints, which are in all the source material that I looked into and everything that the paleoanthropologists described it as being indistinguishable from modern human footprints. So
100%. I have got to get it on this, because I had a conversation. I used to get coffee with a young Earth creationist friend of mine who was a coworker, and we had a very lengthy conversation about the Laetoli footprint. So I am on top of this.
Well, before you explain it,
sorry. Go ahead.
Okay, so you would agree that they do look exactly like modern human footprints, and they were were these discovered or dated at about 3.5 million years ago in actually, where were they? Where were they discovered?
Tanzania. They were found in Tanzania. They're the Laetoli footprints. So
right, the Laetoli footprints.
Yes. So as far as them being indistinguishable from humans, I'm going to tell you about a very cool study that you should look into, because I found it fascinating. Yes, they do look very similar to an organism that has an inline big toe. Now, if you look at the this is the difference between these and the Crete tracks, because you can see the individual toes on some of these prints are incredible. You'll see that, no, the toe isn't completely in line. It's just very, very close to being in line. So what the researchers of that project, of this paper that I'm about to tell you about, did was they were like, all right, well, let's take casts of it, and then let's see if we can figure out how whatever was making these prints bore its weight. Because essentially, as I'm sure you know, the biomechanics behind walking on 2 feet are very, very specific. It's, there you have to have, proper weight bearing on the knees, and you have to have a specific arch in the foot, which is great because you can tell from prints how something bore its weight. So what they did was they had a bunch of hunter-gatherer individuals from Africa. Someone similar to like the Hadzabe. They had a couple dozen of them walk in soft sediment, and then they took a cast. Then they had some chimpanzees walk across soft sediment, and then they took a cast. And then they took the Laetoli footprints. And did you know that the individual that made the Laetoli footprints bore its weight in a perfectly intermediate fashion between the two? And the researchers who published this paper said it is absolutely certain that whatever made these tracks was not a human and it was not a chimpanzee. And they're not even certain, I believe, that it was afarensis. It could have been a.
Well, did Mary Leakey? I'm sorry. Because I'm trying to follow you. Mary Leakey, the one that discovered these footprints 25 years ago, and she admitted that they look nearly identical to modern human footprints. I understand you're saying that there's a few differences that suggest otherwise. Did she conclude that it was from some type of hominin, or did she conclude that it's from some type of Australopithecine? Like, what was her conclusion on it?
She concluded that they were from afarensis because they have afarensis feet, and the toe is close to being as in line as an anatomically modern human.
I thought that was Donald Johanson that concluded that, because I know that he concluded that all these different bones were.
Well,
just to be sure that in case she wasn't finished.
Yeah, well, thank you, James. But I'm sorry, I do tend to go on monologues, so feel free to jump in if I kind of get going. I just get really excited about this stuff.
I definitely like to see, the data on that. It's my understanding that Mary Leakey did disagree with Donald Johanson and him concluding that all, the footprints, all the different bones that were discovered in both her area and his area were actually one species. I do want to, because we were talking about the trackways for a while, I did want to ask you though, regarding, because if we're going to talk about human evolution, right? I understand that you would believe in, would you believe in the out of Africa theory, or would you hold to more the multi-regional theory in regards to Homo erectus?
I think it's kind of important that we wrap up that past conversation, because yes, Mary Leakey and Johanson had different opinions, but the species, as if I remember correctly, and as far as the people go, I can get kind of hazy. There are a lot of people involved in the community, but at least as far as I remember, the dispute was not between whether it was afarensis or a modern human, but whether it was Australopithecus afarensis or Australopithecus anamensis, who was also in the location and has also been at least purported to have. I don't think we have the feet of anamensis in the same. We have a lot better representation of Africanus and afarensis than we do anamensis. But essentially, the dispute was not. I feel like that's a bit of a mischaracterization, right? Because it's not like Leakey was saying, "Oh, they're so close to human; they're definitely human." She was just noting how incredible it was, because the dispute that was going on at the time in the anthropological community was whether or not our ancestors, at least according to evolutionary theory, got big brains first or started walking on 2 feet first, because this would very much be indicative of a bipedality first stance. That's why she noted that they were so human-like, because it was a big find.
Right. So I do understand and agree with what you're saying, that there were disagreements. I was under the impression, I believe, that she definitely didn't at least designate it to Donald Johanson's Australopithecus afarensis. And I know that there were some disputes whether or not all those bones, the footprints, did they belong to one species or was it multiple species? But at the end of the day, if the evolutionist does have some type of explanation regarding the modern human footprints found in Laetoli or found in Crete, I just find it funny, because evolutionists are always asking you, where's the evidence that Homo existed in these layers with our so-called ancestors? And if we point them to, say, footprints or some bones that are indistinguishable from, say, the Homo genus, then usually there's a circular argument that has to explain it away, instead of just admitting, okay, there is something that looks like Homo existed during this time. So it just seems like there's a circular argument, because the evolutionists can't admit that, yeah, these are human-looking footprints, because they don't believe that humans lived during this time. So it just seems like a circular argument. I'm not sure if it's something that just needs to be agreed to disagree upon regarding the footprints, but I definitely like to see the paper that you talked about regarding and if it was indeed not a footprint, because I haven't seen that, and then also the information on Leakey and what she concluded.
let me send you after this conversation. I will send you. I've got some awesome papers just like on the back burner that I can kind of just send to you so you can look through them yourself. But
Awesome.
it's not so much. You mentioned that it's like show me the evidence, and then we find what a potential footprint in Crete, and then human-like footprints in Laetoli. Well, no one ever thought that those were human footprints. I would challenge you to find anything by Mary Leakey saying, yes, they are certainly human footprints, or I think that they're human. Not, wow, they look really human, because they look very human, aligned with one of the primary hypotheses at the time, and it turned out to be the prevailing one. That's what we abide by today. When we look at the hominids, we see all the traits for bipedality show up way before the brain cases start exploding in size. So I don't think that it's people ignoring the evidence. This is just the natural progression of things. The Crete thing, yeah, I can send you what I got on that for sure. Because I gotta say, if your evidence that there are hominids living in Europe, and that one of them made the Crete footprint, if your evidence for that is something that may be a footprint and may not be, is all you got, considering how well things fossilize all the time elsewhere, which isn't to say they fossilize very easily. I'm just saying, to find no trace of any hominid there, rather any Australopithecine and back hominid, I don't find that particularly convincing.
Right. It just seems based on findings all the time there is. I mean, even now they agree that there's more of a bush instead of a tree regarding the, ape to human evolutionary ancestry. And then regarding certain things like fossil footprints or Crete, from what I read in the primary, source data, they are concluding that they are footprints, but, who actually made them. So I think it just comes down to the fact that I'll have to look at, the paper, the material that you're suggesting. That maybe they're not human footprints, but I think even moving away from that topic though, because I am really genuinely curious if there is any validity to human evolution. I think it's reasonable that, especially if you're in the affirmative, I've always struggled with the evolutionary out of Africa theory, because it sounds like it really does involve an extended near extinction event, but that extinction event would be associated with severe inbreeding. So for one, do you hold to the out of Africa scenario or the multi-regional scenario, and kind of go from there and maybe give an explanation as to how that might be feasible regarding Homo erectus and then Homo sapiens?
Well, generally speaking, at least as far as I know, the current consensus on things, because as far as what I think, I try to go off of my impressions of the current literature. And the literature changes, because you're right, that is the nature of science. Like the time scale has been swapped around several times in light of new information, which is kind of how science should work, at least in my opinion. You always want to be closest to the truth. But things have been changing less and less all the time. Now it's slight tweaks here and there. And as far as I know,
Right.
the general trend of the gradient of hominids has gotten pretty solid. I don't think there's been a large change in that in quite some time, other than the finding Homo sapiens quite a bit older. I think it went from 200,000 to 300,000, which is very neat. But I digress. As far as the out of Africa theory and multi-regional, I think what's currently going on, and that's not my specialty. In fact, I found that part slightly less interesting than the hominids and the morphology itself when I was taking human evolution. But I think the current consensus is that there were, I think, 3 kind of bursts where you had erectus leave, right, and diversify into Floresiensis and Denisovans somewhat far away.
Yeah, right. I believe even and sorry to interrupt, I believe in the
No,
out of
it's
Africa
okay.
scenario, there was 2 out of Africas. So maybe a couple million years ago, after Homo erectus evolved, Homo erectus left the continent of Africa, and then independently on the other continents evolved into, for example, exactly what you just said, right? The hobbits.
Right. Are you
the
certain though
Denisovans.
that it wasn't Heidelbergensis that diversified into Neanderthalensis outside of Africa, into Neanderthalensis? Because at least what I am aware of is that we share a common ancestor with Neanderthalensis in Heidelbergensis, not Erectus.
Well,
So
right,
then
you
it
got
would,
Homo heidelbergensis, Homo
right. So then it would
neanderthalensis.
stand that there would be 3, right? There would be one where Erectus leaves and you get Floresiensis and Denisovans, and then there would be one when Heidelbergensis leaves and you get Neanderthalensis, and then Sapiens leaves as Sapiens, from Heidelbergensis, but staying in the African continent.
So as all these hominins are evolving, right? So Heidelbergensis, Homo neanderthalensis, the hobbits, Naledi, obviously the Denisovans, for example, and then you've got Homo sapiens that are in Africa that evolve out of Homo erectus. They, according to what I've read in the primary literature, they would have had their migration, but that was also after the fact that they had a near extinction event, because we know that every single human being on this planet is no less than 99.9% similar, right? We have low genetic diversity.
Totally.
From my understanding, I'm almost done. From my understanding, the evolutionists were kind of caught off guard regarding that. So that's why they resorted to a population bottleneck roughly 70,000 years ago that would have reduced homogeneity, right? But my question to you is, how could such a tiny, nearly extinct, obviously, Erica, they'd be genetically compromised due to inbreeding, prolonged inbreeding, suddenly explode into all parts of the planet like you talked about, and then seizing dominion over the world, whether they assimilated with all these other hominins, you know, Heidelbergensis, hobbits, Naledi, for example, which obviously hobbits and Naledi, I conclude are just humans that are genetically compromised due to inbreeding and isolation post-Babel. But if there's any validity or feasibility to human evolution, how do you explain those humans diverging from Africa, even though they should be so genetically compromised they should have gone extinct? Go ahead.
Well, no, not necessarily at all, actually, because the bottleneck that humans are thought to have experienced due to the Tobu eruption.
Right.
Hold on.
Yeah.
Just to be sure that. Okay. Go ahead.
Yeah, so the humans experienced, right, a bottleneck due to the Tobu eruption. Now, do we know of any other organisms that have experienced an extreme population bottleneck? Yes, we do. Everyone loves cheetahs.
Right,
cheetahs are essentially genetic. All of them are very close to being genetic twins to one another, which is indeed the two of them are much closer to one another, take any two random individuals than any two random humans. So I would say, yeah, it looks like they did an A-OK job recovering from an extreme bottleneck that was much worse than that of humans. My question to you would be, in response to that, because my point being, I don't think it's any problem having a population bottleneck. I'm fairly certain that the population wasn't even that. It was pretty small, but it wasn't that small.
How small would you say? I'm sorry to interrupt.
Before we go out on that rabbit trail, just to be sure that that last point is wrapped up.
Could she just say, is it 2,000 or is it 10,000? I've heard some evolutionists say 2,000 people, other evolutionists say 10,000. I mean, what do you hold to?
I don't know. I don't know the literature on that. I know about the Toba extinction, but that's not, like I said, out of Africa and that particular bottleneck aren't my specialty. I could talk about morphology all day.
Hold on, Standin, just to be sure that with that before we move on to in case you didn't finish that last point, Erica.
Thank you, James. I was just going to say I don't know for certain, and that my kind of reverse question to you would be, if you're pointing to the bottleneck as being a result of kind of the flood, my question would be, all right, why don't all species have such a severe bottleneck? And why do the cheetahs also have a bottleneck? Why is it humans and cheetahs? You know, what's the model for that?
Okay, very good questions. I'll answer those questions. I want to kind of object to what you said. I find it funny every time I ask this question to evolutionists, I do find that they fail in answering it, because they always bring up the 10,000 cheetahs that we see in the world today. We know that conservationists, do feel the cheetah is already showing serious signs of inbreeding and genetic decline. I mean, there's just not enough of them. And it's a fact that genetic diversity in these cheetahs has eroded. And what they're seeing now is the expression of many destructive mutations. So all the answers that I get and that I find in the primary literature regarding this out of Africa are not credible, because small bottleneck populations have extreme problems. And the thing is, this bottleneck would have been prolonged, due to the inbreeding, due to the genetic compromised population of 2,000 or 10,000, they would not have been able to seize dominion over the world. That's a fact.
So why though?
Okay, unless you want me to answer. So was your question how do we?
Oh, sorry. Right. Go ahead. Yeah. Go ahead. You can. Yeah, but go ahead. That's fine.
Okay, so why is there not? Actually, if you can rephrase your question. So is there a problem with our bottleneck after the flood? Is what you're saying, like due to inbreeding or? Well,
I'm saying if you've got if you have a single pair of multiple individuals, you should have every single organism that resulted from those kinds essentially looking like the cheetah right now. Much worse, actually, if you're going from a single breeding pair. I mean, we consider that functionally extinct in ecology today. I mean, it happened with the northern white rhino. That is an unrecoverable loss of genetic diversity. Because you're right, genetic diversity absolutely matters. The cheetahs are experiencing trouble, but I don't know that I would phrase it as that they're experiencing, a higher rate of mutation. The problem is that their lack, I mean, maybe I don't know enough about genetics to say, but as far as I know, I thought the primary problem was that they're all so identical that if there's a single disease that hits them, they have no defense because there's no kind of variety, which is really important. But yeah, that was my question.
Yeah, and I think it all comes down to the fact of where does genetic diversity come from? Because the big difference between my view and then say your view, Erica, was that I explained the vast majority of DNA differences, right? For example, nuclear DNA differences, not by mutation. And that's what evolutionists explain them by, right? All the millions and millions of DNA differences they say are the result of mutation. But according to our model, we explain them by pre-existing or created DNA diversity. And that one genetic difference between your model and my model has massive implications for the timing as to the origin of species, for example. So if we have these differences from the start and therefore no mutation accumulation by the time we reach the flood and the population bottlenecks, for one, very little heterozygosity regarding the original created heterozygosity wouldn't have been lost. There wouldn't be any bottleneck problem. We wouldn't expect to see the same thing that we see with the cheetahs. But regarding your model, you guys do explain the vast majority, if not all, the DNA differences as the result of mutation over time. So mutation accumulation and the results of inbreeding pretty well makes your out of Africa scenario impossible. And
well, I don't see why though.
Reasoning, but nothing really that's feasible or valid. So that to me destroys the model. Go ahead. Take it.
Sure. Sorry. Yeah. I was just going to say, I struggle to see why you're having a problem with that. I mean, cheetahs aren't doing the best that they could, but they are certainly not doomed to extinction, and they have a much tighter population bottleneck than that the ancient humans are proposed to have had. So I don't understand why you're saying, I mean, the cheetahs did it and they're still doing it, and the humans' problem was less. So I don't see kind of like, I feel like you're kind of saying, oh, it's a problem, but then it's like, why is it a problem? You're not providing a reason other than saying, well, it's bad, there'd be inbreeding. Well, there might have been, but it clearly wasn't too much of an issue. I mean, that's kind of like,
well, exactly why you say it must not have been an issue, because you have to hold to that primary assumption that ape to man evolution actually occurred. But the reason why is because that evolutionary bottleneck hypothesis, like I said, it involves an extended, that's the problem. It's extended. It's not one generation like our bottleneck. It also involves a lengthy near extinction event. So that is a fairy tale, because that hypothesis is associated with severe inbreeding is not even remotely feasible. What you said about the cheetahs, yeah, conservationists are trying to keep them alive due to the recessive mutations that are being fixated in the populations. I mean, their sperm is degenerate. They probably will go extinct. They would have gone extinct if it wasn't for conservationists. But this,
not at all.
2 to 10,000, well, this 2 to 10,000 population of Homo sapiens that came out of Homo erectus would not have been able to seize dominion over the entire planet. Plus, there's evidence that all of these populations you're talking about, right? I liked your analogy on your last debate about Middle Earth, because I love Lord of the Rings. Yeah, it was kind of like that. You got Neanderthals, hobbits, Naledi, Heidelbergensis. You've got the Denisovans. You've got them all living at the exact same time.
So I guess we can just kind of leave it at agree to disagree. I don't particularly see a problem being, with such a bottleneck, considering that we've seen others. I would like to, if it's okay, since I would like to maybe take a turn picking a topic, I would like to talk about Chromosome 2, because you brought that up in kind of a, well, it's been overturned. I would say not in the slightest. Let me fix this here. My thing fell.
I just want to ask the moderator, because, yeah, obviously I want to talk about Chromosome 2 fusion. I know we had a 30-minute discussion. I want to know how much time we do have to talk about Chromosome 2. James?
I've got flexibility with time if you guys do, but that's...
Yeah, I could go all night. I'm good.
So I think we've got enough time to go, We go for another 45 minutes, including Q&A, like which would be like 25 minutes at the end, maybe even 30. So I would say if we go maybe for discussion wise, maybe another like 15 minutes, and then go into the Q&A.
Sure.
Sounds
Perfect.
good. Yep. Okay. So what
Okay,
would
so
be? Yep. Okay. Go ahead with the
let's
chromosome
talk
2
about
fusion. What
this.
would be your objection, I guess, to the claim that I made overturning it?
Okay, so, well, this would be kind of it, because Jeffrey Tompkins is known for being the guy when it comes to debating the human similarities with chimpanzees, which I did look into that 88% claim, and it is somewhat, I hate to be kind of sassy, but it's very abysmal. First of all, he used an extremely buggy version of BLAST when he got that first 70% number, and no one can seem to get that 88-ish% number when they're running BLAST how it's run in every single other species. It was kind of, as far as I know, it was revealed that he was essentially saying, all right, if you have a strand of DNA and there's a single base pair difference in the middle, that goes from being 100% identical to being 50% identical, which is very interesting that he's choosing to run it that way with humans, considering we don't run it that way with any other species. But with regard to human chromosome 2, essentially Tompkins argues mainly that it's not a fusion site because there is a functional pseudogene at that site. Now, first of all, it would be functional in that it is indeed expressed. It's barely expressed, first of all. I mean, it's barely interpretable from noise in the background, but yes, it is there. But there's also a promoter.
Okay, wait a minute before you go on too long, because I just want to touch on that part that you just said.
Sure.
Because you're right. We do look to that reputed fusion site, and we're saying that it's overlapped by a functional gene, okay?
Yeah.
So that so-called fusion site is, we're arguing that it's not actually a fusion site at all. It's indeed a functional component of that DDX11L2 gene. But are you aware that that so-called fusion site does lie within the first intron of that gene, and that it also is an active promoter for the transcription of an alternative transcript, Erica? It also binds at least 12 transcription factors, and it initiates RNA transcription, which begins with its own sequence, and it has a chromatin profile that further supports its active promoter status. So why do you say that there's barely any function and barely any activity there, when these are the facts that we understand?
Well, you say all those things as if they're this grand and vastly vital process. But if that were indeed the case, and I'm not a geneticist, so I don't know this for certain, I would imagine that it would show up as more than just noise in the background. I mean, you had to scrutinize that this portion of the genome to essentially even find that there was a pseudogene there. And furthermore, are you aware, Standin, that there are the calling card for telomeres right at that fusion site, constantly and consistently repeated, this both the forward, which is TTAGGG, and of course the reverse, which is CCCTAA, and they're flip-flopped just like they would be on the two opposite ends of a telomere. So I don't, and as for there being a functional gene, it's also been.
Hold on, one second. Let's just.
It's just because genetics.
Hold on, I know. Just to be sure that we get plenty of time, we'll let her wrap up. If you can wrap up this point, Erica, and then we'll kick it back.
Yeah. Sorry. Yeah, I was just going to say, furthermore, there just being a pseudogene there that is indeed functional. We see functional pseudogenes on fusion sites in other animals that have been accepted to have had chromosomal fusion, even by creationists. This is a paper that was done on equids, where it does indeed happen. So I don't, and I can provide that paper for you too if you'd like. But essentially, as far as the conventional science goes, there is like actual empirical evidence that promoters can cause pseudogenes to essentially relocate onto a fusion site. So I don't understand, essentially everything rests on this being a functional gene, but that's not problematic. It's been shown in other animals to be A-okay.
Well, I don't think it's just because of that, because you did talk about the telomeres, right? The ends of chromosomes. And yes. I'm sure you would agree that telomeres, they are served in our genome as end caps that actually protect the DNA, and one of their jobs in protection is to prevent fusion to begin with. So why were they even involved in this so-called reputed fusion if one of their jobs is to actually prevent it? And from my understanding, this apparent hypothetical fusion, which obviously we can't observe happening, two questions. Why can't we reenact it in a lab today? We don't have any DNA from fossils to see, how it happened, if it actually happened. And lastly, it's not really evidence for ape to man ancestry anyways, because even if I conceded that there was a chromosomal 2 fusion, as you know, we've got the chimp and human ancestor who split. And then in the human line is when this supposed chromosomal 2 fusion occurred. So either way, it's indirect evidence for ape to human evolution. So that's no help to you. And secondly, we have all these discrepancies showing that possibly it isn't even a chromosomal 2 fusion site to begin with. So it's kind of like 0 for 2 for the evolution.
Well, but the problem for that is there aren't refutations. Like I said, this is a known phenomenon. I mean, this paper was in 2013. We've known this for 6 years, that this fusion sites can indeed hold functional pseudogenes on top of them too. Why it's important is because creationists for the longest time were like, well, there's a chromosome difference. They can't be related. Well, yeah, they can, because what if the chromosomes fused? And look, they did. And this was a prediction that was made and, met. And the only as far as what you've said here and what I've seen from Tompkins, the only essential refute isn't even a refute. It's basically a guy saying, well, I have a problem because sometimes pseudogenes are functional and there is indeed one on top of the fusion site. But then here it is, not problematic in equids. And oh, sorry, go ahead.
Is this specific telomere to telomere fusion? Is this of its kind? I know there's been other chromosomal fusions, but this specific one that they're purporting, is that the first documented type ever seen in nature?
I wouldn't imagine so, since it happened in the equids.
Right, but the one in the equids, I believe, and I don't have it with me, the paper, I believe it was a totally different chromosomal fusion than the one that's being purported here, right? This telomere to telomere fusion. You didn't really answer my question regarding telomeres anyways, how they're used to protect the DNA, and one of their main jobs is to prevent fusion. So why should we believe that they were involved in a fusion? But this is a hypothetical fusion too.
Sure.
We see these chromosomal fusions happen in nature that I believe aren't the same as this reputed one. But why are we to believe, just like the out of Africa scenario, which I don't believe you answered as well, which kind of destroys human evolution, why are we to believe that actually occurred? Because it's all theoretical, hypothetical.
The human too? Well, here, I'll let you pick. Would you rather me answer out of Africa, which I thought I did, but maybe I didn't. I might not have, genuinely, I can't remember. Or would you rather continue with human chromosome 2? You picked the path.
Yeah, you did answer the out of Africa, but kind of like you accused Hovind of answering your question, but then you said that, he didn't really answer it sufficiently. That's what I'm saying about the out of Africa, because there really is no feasible way for this 10,000 Homo sapiens to seize dominion over the entire world after a prolonged bottleneck of severe inbreeding, the accumulation of mutations. I mean, it's just not feasible. It's fairy tale. When it comes to the chromosomal 2 fusion, I just want you to answer the question. If telomeres, one of their jobs as being the end cap of chromosomes, is to prevent fusion, why are us to believe that somewhere down the line, millions of years ago, after the split between chimps and humans, this purported fusion actually occurred, and that it's unlike anything we've actually seen in nature regarding fusions? I mean,
okay, hold on. Just so we got to wrap up this point, standing for truth, just so we can kind of keep it going back and forth.
Yeah, sure. Standing, you want to finish that, or you want me to go ahead?
No. If you just want to, I guess, answer that question regarding the telomeres.
Yes. Yeah. So with telomeres, why are there mutations at all? I mean, our DNA, when it's actually replicating, there are checks and balances that are supposed to prevent mutations from happening that fail all the time. That is the nature of genetics. So telomeres are supposed to protect from that, but it obviously doesn't do it all the time. Now, as far as this not being the same kind of telomeric fusion, I'm not sure. I'm pretty sure it is when I kind of eyeballed the abstract, but I can't say for certain. Genetics is not my forefront. But I would like to talk about out of Africa, because I do know enough about genetics to say that we have a real-time example. I keep bringing up the cheetahs because I'm not understanding why it's problematic for humans. But
I don't want to interrupt, Erica.
Hold on.
I want to go back to the
one sec. Just to be sure. Okay, hold on one sec. Standing for truth. Just to be sure that, because want to be sure that she's got plenty of time to respond, because I think there were two questions.
I'm just trying to answer.
Oh,
both,
right.
you
You're
know?
right. You know what? I apologize. I did ask the out of Africa one again. So
Yeah. I'm just trying to get them both in.
chromosome 2.
Yeah. I'll quickly just rehash out of Africa, and then that's kind of why I was like, let's pick one. But we can try to juggle both. That's fine with me. With out of Africa, right? I mean, so I'm gonna really just like make my point as kind of concise as I can. Humans have a bottleneck. Cheetahs have a bottleneck. The cheetah bottleneck was much worse than the human one. And you don't have to be there to know that, because all cheetahs are more closely related to one another than all humans are today. So know that they're less genetically diverse because of a bottleneck. Okay, great. Cheetahs did very well for themselves until humans started hunting them and destroying their habitat, which are the main reasons why there are less of them today than there used to be, not because of these new genetic mutations popping up. As far as I know. Anyways, I'm not a cheetah geneticist, although that would be a cool job. With humans, it shouldn't be any more of a problem. I mean, essentially, cheetahs had a worse situation and did just fine until something completely unrelated hit their numbers hard. So with the humans, and also, mind you, the Toba extinction, or the Toba eruption, was indeed a volcanic eruption in nature. So while, yeah, I think that the estimates are around 10,000 individuals, that isn't certainly the case that there weren't other bands of humans out and about. Now, granted, it probably wouldn't change that number very much, because as you know, genetically, that number is genetically kind of constrained for a reason. But you're essentially saying it's impossible, at least this is what I'm gathering. You're essentially saying what my ears are picking up, that humans had a bad bottleneck, thus they shouldn't have been able to spread out and essentially diversify across the planet. Except cheetahs had a worse one and they diversified across Africa. So that's my answer to out of Africa. I don't see it as an issue. I don't think really anybody does. I've actually never heard that argument before. And I look at a lot of creationist arguments. Anyways, that's my answers. Those are my answers.
Awesome. Okay, if I could just say a few things. So the cheetahs, for example, are being looked at from conservationists today. Inbreeding is the issue. We know inbreeding exposes the genetic mistakes within the genome. And I always say regarding genetic entropy, inbreeding is like a sneak preview of where we are going genetically as a species. I mean, we accumulate 100 new mutations per person per generation, most of them being nearly neutral. What we now know about the function of the genome, that means we're degenerating faster than we ever thought. Now, this population bottleneck in the out of Africa scenario was conjured up by evolutionists once they realized that human diversity is very, very low, indicating that we must have come from a very small population. That small population, according to my belief, obviously is Adam and Eve. That makes sense. God created Adam and Eve. What do we see today? Low genetic diversity. Me and some man over in Asia, no more than 99 or no less than 99.9% similar. That makes sense. Evolutionists has always had to come up with a rescue device. Yeah, there must have been some kind of genetic bottleneck 2,000 to 10,000 Homo sapiens, say 70,000 years ago. The thing is, this was a prolonged bottleneck involving severe genetic damage due to inbreeding. Because when you get small populations like that breeding together, not for one generation, not for 10 generations, but many generations, as the out of Africa scenario would suggest, that that population would have been so genetically compromised that there's no feasible way that they could have seized dominion over the entire world. So I'll let you comment on that if you want. But the chromosome 2 fusion, I just want to, with the chromosome 2 fusion, you were talking about the telomeres, and we should see remnant telomeres. But from my understanding, there's, for one, a struggle to find these. That area is very, very degenerate. Why aren't there any more sequences? I think it's not just the fact that the purported fusion site is actually a functional DNA element in a human gene. There's other discrepancies that go along with it. So not only that, not only the telomeres, but also the fact that remnant centromeres are also difficult to find. So it's not just the fact that a gene has been found and it spans the fusion site. It's a number of things. But like I said, it's still indirect evidence because this supposed chromosomal 2 fusion occurred in the human line, which means we could easily explain it based on either pre-flood or post-flood, especially because we believe in a population bottleneck anyways. It would have been passed on to everybody we see on the planet. So it's just not very good, convincing evidence anyways for ape to man evolution. I just think you're going to need something better. The pseudogenes you talk about, we know that they have very important functional roles in immune response. They're necessary to sustain healthy life processes. So go ahead.
Yeah, sure.
Actually, take as much time as you want, Eric. I talked a lot there.
Yeah. Well, I'll introduce something quite interesting in a second that you've heard before. But one, the reason we do find remnant telomeres, this is actually Tompkins's research here. So those are the remnant telomeres that you're looking for in that central part of the chromosome. With regard to, kind of pushing an extinction event, we know the Toba extinction event happened, I'm fairly certain, because there are ash beds. There are enormous ash beds, and that's how we know that it was a really bad extinction event, or really bad eruption, sorry. And really bad eruptions tend to not be super good for the things that are indeed around them. We know that from all the volcanic eruptions that we see today. That would be my sort of answers to that, but to kind of take a swerve, and feel free to answer those if you want. I mean, I think we're just chasing our tail with that. We're just going to disagree. But I'll ask you the same, you brought up my question to Kent, so I'll ask you kind of the same question. If we use genetics, right, to determine relatedness within the human population, why can't we do it between populations? Because I find that quite compelling. You don't find that as genetic evidence very compelling. I was mostly using it as a refutation, because creationists often say, oh, well, there's a chromosomal difference. Okay, well, it's a fusion. But the fact remains that that's not really the point. And it's also not just the fact that there's a fusion, but that the tops and bottoms of that chromosome are very, very, very similar to 12 and 13 in genus Pan. So where do you draw the line? I asked Kent. I didn't really get an answer from him. So where do you draw the line?
So where do we draw the line on common ancestry?
No, where do you draw the line between humans and chimps? Why is it okay to use me and my mother's genome to confer a common ancestry between the two of us, but not between me and a chimp?
Right. So if we're looking at, for one, we'd have to look at DNA differences. It also comes down to the fact that humans, okay, for all of recorded history, have produced humans. And we can use mitochondrial DNA, or we can use the Y chromosome DNA, and we can test whether or not, or we're related to our father. We can actually prove that the entire, all of humans, the entire human species are related. This is verified through independent corroborating evidence. Okay, but now you want to extrapolate that to pretty well suggest that humans and chimps are also related, even though there's so many discrepancies. Because I always say there's no point in arguing over evidence that's kind of agnostic to the debate. And what I mean by that is genetic similarities, whether it's nested hierarchies in DNA on a molecular level, mitochondrial DNA, for example, nuclear DNA, or nested hierarchies regarding anatomy, morphology, physiology. This can either be explained by common design or common ancestor. We need to look for evidence that can differentiate between the two. And I believe there's plenty of lines of evidence that can differentiate between the two. For example, the Y, just one sec, the Y chromosome. Yep. Okay. So this is going to be my follow-up question to what you said. Let's look at the Y chromosome. Okay. Evolutionists were shocked. The Y chromosome is the most stable chromosome in the human genome because it doesn't exchange its genetic content with anything else. It doesn't experience recombination. Yes, gene conversion, of course, amongst itself, but this should be the most similar chromosome in the entire genome regarding apes and humans. Why is it less than 70% similar?
Am I good to go?
Yes, take your time.
Okay. So I feel like that's a little bit of as Kent would put it, like a hit and run, right? A slap and run, because that didn't really answer the question. I mean, you basically said humans produce humans, therefore it's reasonable that genomic comparison between me and my mom would be common ancestry. Okay, well, what if you had an individual, an orphan? You don't know where they came from. You didn't witness their birth, but they certainly have a similar genome with the mailman. You didn't see it. You don't know for a fact that is indeed the father or the mother of this child, but their genome is very similar. So similar, in fact, that it mimics that of mother and offspring, or father and offspring, in other groups. So I think you would indeed point to that and say, okay, well, common ancestry. And that's what creationists do within their kind groups when they're formulating their kind groups. My argument with this isn't necessarily to say, whatever, pine trees and elephants are related, but to point out that there's no way of getting around lumping humans with the apes. Because we use in a court of law genetics as a means of relatedness. I mean, you're basically saying we should draw an arbitrary line because we have never seen a human produce a non-human, which even if they did, that would violate the law of monophily. But yeah, because recorded history has been around for like, what, 10,000 years? Less? And even then, it's barely been recorded. I wouldn't suspect no one under an evolutionary lens would suspect that you would. So I feel like that's somewhat of a non-answer. You showed like the transitional designs earlier. Okay, well, where's the plane with wings that can't let it fly, or with half of an engine that performs a different function? I mean,
okay, here, let me respond to that real quick. I think you went.
Yeah, sorry, I did dodge around.
Yeah, no problem. So here's my answer. That's not quite as technical as what I said regarding genomics and genetic data that
Sure.
can differentiate between the two. For one, as I've stated, we would expect nested hierarchies, whether it's anatomical, morphological, physiological, or genetic related hierarchies. But regarding your question of where do we draw the line, why can't we conclude that apes and chimps are related, but we can conclude that me and my grandmother is related? Well, the thing is, we need independent corroborating evidence. Okay, so not something that also doesn't rely heavily on assumptions and inference. That's what I'm saying. There's an extrapolation fallacy there. So here's one example before you go. We have DNA testing, like you're talking about, that confirms, okay? There's no argument. Nobody argues, I hope not, that all humans are related, okay? Also mother, father, grandmother, we can see this based on maternity tests, paternity tests, but these various testimonies are completely distinct from DNA, okay? There's also corroborating, because we validated this test over and over and over again in humans. Like you've said, all of recorded history, humans have produced humans. But unfortunately for the evolutionists, okay, now you want to extrapolate that to mean humans and chimps are related. Well, we can't go ask the chimpanzee, we can't go ask animals from millions of years ago, or even animals from today, for their testimony regarding the DNA testing of them and their offspring like we do with humans. So there's just no way for us, Erica, to have independent corroborating evidence here regarding your question. But we do have independent corroborating evidence when it comes to all humans being related. You're committing an extrapolation fallacy.
Just to keep it short and pithy, standing for truth.
Yeah, 100% agree. We do know that all humans are related. Why do we know that? Because our genetics are so similar. So, well, why do we know that humans and Neanderthals are related? Well, because their genetics are so similar. You can take that back down the line, but creationists can't come up with anything empirical to draw the line. You say, well, we don't see it. We can't ask a chimpanzee. Okay, well, we can't see the inside, or rather how a black hole functions in real time, but we can make accurate predictions about how light's going to react around it because of the data that we do have. So there is no reason why you can't extrapolate it backwards, any more than there isn't a reason why 23andMe can take your DNA and take your DNA and extrapolate backwards and say, guess what? You're 63% Norse. Well, why couldn't they? I mean, you're essentially kind of saying that this is impossible because no one was there. It's an appeal to observational science, which I'm not a very big fan of. And then you say, where's the corroborating evidence? Well, it's all the other stuff I showed. It's the fact that humans are morphologically nearly identical in so many ways to all of the other apes. It's the fact that we have this smooth gradient, and creationists can't come up with a meaningful way, at least in my opinion, to say that we can have this accelerated decay that completely throws 4.8 billion years out the window. It's the archaeology, the fact that no one can explain why there's so many dang tools all across the surface of Africa. All of that points in one direction, and it isn't to 6,000 years. So it's kind of like parsimony, right? What's the most parsimonious explanation? Because I'm not going to sit here and tell you that we know for sure exactly how things went, because we don't, and I don't think we ever will. I would love that. That would be awesome.
Okay, can I respond to a few things that you said?
Yeah, okay. Sorry, I'll wax poetic. I'm guilty of the same thing.
No, I think we might be both guilty regarding that. So I'm not saying that it's only an extrapolation fallacy that we can look at similarities in our genetics. Like I said earlier, every single human being on this planet is 99.9% similar, which says we have low genetic diversity, indicating we came from a small population. Okay, this is a good thing for us. Now, you want to extrapolate that to mean we also have similarity with chimpanzees, for example. And then we also have similarities with monkeys. We have similarities with dogs, whales, fish, all the way down the line to a bacteria, the nested hierarchy of life. I'm saying we too predict nested hierarchies. Therefore, it's difficult to draw that line since we both predict the exact same thing. We need something that's going to differentiate between the two lines of evidence that both models can explain. That's why I pointed to something, because you talk about similarities. The Y chromosome, for example, and this time hopefully you can answer it. But the thing is, the low level of recombination and sequence diversity on the Y chromosome that I'm talking about should make the Y chromosome the most similar between humans and chimps. But the problem is that the human and chimp Y chromosomes are actually incredibly dissimilar when they should be the most stable, because they are immune for the most part to recombination. So how do you explain the fact that the Y chromosome is less than 70% similar between chimps and humans?
Well, first of all, I would, as I've continued to kind of begrudgingly admit throughout the course of this conversation, that genetics really isn't kind of my field. I can talk to you a lot about, other stuff, but genetics isn't it. So I'm not kind of adept enough to really answer your question in a way that I feel that you would find sufficient. I don't know. I would ask you out of curiosity if by chance that 70% number comes from Tompkins, considering Tompkins.
Nope. I've had a good question. I have the paper where it came from.
Do you know who the author is?
Source data, because they were shocked by it. But from my understanding, talking to scientists, right, I've debated Dr. Stefan Frello, for example, their explanation would be gene conversion, because it can exchange genetic material within itself, right? It's like recombination is like shuffling a deck of cards, but the Y chromosome doesn't have a counterpart to do that too. Therefore, they should be the most stable. It's the best chromosome to look at. My question would be to the evolutionists like Dr. Stefan Frello or whoever, Herman Mays, that wants to look to that as a rescue device. What testable prediction can you make and bring forth regarding change on gene conversion in the Y chromosome? Because and it's okay that you don't have the answer to it, but that would be a differentiating line of evidence that can tell us, are chimps and humans actually related? There's no way they can be, based on the dissimilarity of the Y chromosome. Less than 70%. Evolutionists were shocked, Erica. Shocked.
But here's the problem with that. That's kind of essentially hinging it all on the fact that I don't have an answer for the Y chromosome. Now, what I do know about genetics would be, even if the Y chromosome were 70% dissimilar, well, there are some proteins between humans and chimpanzees that are like 50% similar. And the reason that they're 50% similar is, as we know the mechanisms of genetics can kind of swap things around. But the fact remains that we are still the closest related to chimps with regard to that single protein, even if you're just comparing that single protein across, the animal kingdom where that protein has been kind of examined, would still be the closest to the chimpanzee. It might be only 50%, but you're not going to get any closer. You're not going to find another animal. If you could find a rabbit with a greater similarity, well, then you might have something to kind of go on, unless it was, say, like a remnant that had been turned. If you'd been able to kind of show that it was a de novo type situation, but the fact would remain, I bet. And I don't know this, but I would put a small amount of money on it, because I'm not very wealthy, that the Y chromosome, even though it's 70% similar only, that's still the closest member of the animal kingdom to humans. And it may simply be, as you know, there are areas on the genome that are kind of hotspots for mutation.
Mutational hotspots, yeah.
Yeah, sure, yeah. And they just kind of undergo it quicker for whatever reason. I'm not a geneticist. I wish I could tell you. I wish I knew the answer. But my answer would be, how do we know that something similar is not going on the Y chromosome? Can you show me an animal that's closer than that would kind of violate it? Because as far as what I know about all of this stuff, that's still not enough to say humans aren't apes, aren't a member of the ape kind. Would you be the type of creationist that would say, no, humans aren't primates? Because I know Evolution News admits humans are primates, and I know Creation.com admits humans are primates. Even if they don't like common ancestry, they admit that morphologically and genetically, we should be classified as primates. AIG, they don't like it. But where do you stand on that? Where do you stand on that? Do you think humans are primates? And, if not, why not?
Okay, so first of all, with the proteins and Y chromosome comparisons, before I answer the question, just to address a few things.
I went on for a while.
No, and that's okay. The whole point of looking at the Y chromosome, say, versus the proteins, anyways, is because the Y chromosome is known scientifically to be the most stable of chromosomes. So that would make sense to look at that to see, how close the similarities really are between humans and chimps. But I don't want to beat a dead horse regarding your question regarding ancestry. Anyways, to begin with,
and then forgive me, sorry to interrupt. Not you saying for truth, you didn't do anything wrong. I just forgot to mention
for once
we will go to Q&A pretty quick. So if you're both able,
don't we have a quick closing just to wrap things up?
Forgive me if I forgot that from the email. Was that an email?
I'm okay without a closing.
Oh, deal.
Even just a few minute closing just to wrap things up.
Sure, you betcha.
I guess to answer your question, so are we just going to kind of wrap this part up here and then jump into closing and then Q&A, you were saying, Jay?
Well, I suppose what we could do is, if it's something you can fit into your closing, we go into closings. But if it's something that you're like, oh, this is just kind of really relevant to this point right now, like, we can do it.
Okay.
Just short.
Actually, that's good. Yeah, I can
50.
wrap it up in 30 seconds, and then if she wants to have the last word, then we can jump into the closing. So I think Erica, it all goes back to the question, anyways, as to how we identify common ancestry. Okay, so if we both predict nested hierarchies, then we need to look at something that can differentiate between the two models. Now, we explain anyways the majority of DNA differences based on our model, which does lead to testable predictions that are future and novel, as pre-existing functional DNA differences. Now, we only know very little of the DNA language. We speak less than 1% of the DNA language as far as I'm concerned. And all you got to do, Erica, is look at the history of genetics. I mean, in the 40s, there was one gene, one protein, one function. Science changes nearly every single day. I mean, we see massive amounts of literature giving us unexpected functions of RNA. And we've only functionally tested less than 1% of the genome, because the gold standard of genome testing is gene knockout test anyway. So my technical answer, and to end it quick, I know we're going into concluding statements here, is genome function. Because evolution would say the majority of DNA differences, if not all of them, are the result of mutation. Therefore, we're looking at a lot of neutrality. We're looking at a lot of genomic fossils, right? Pseudogenes, for example, endogenous retroviruses, ALUs. Us, on the other hand, our model's based on pre-existing functional DNA differences. Therefore, we make a very specific prediction that the vast majority of DNA elements, DNA sequences, DNA differences are functional. This is a head-to-head prediction that we can test in the lab and see what is true, common ancestry or common design. That's my answer. Go ahead. You can have the last word, Erica.
Yeah, sure. I'm going to make my last word my closing. It's not so much does genome equate to function, it's genome does equate to relatedness, and there's no reason why that line can or should, rather, without empirical evidence, be drawn. I mean, essentially my whole point of this was that you have several lines of evidence that in and of themselves are convincing, but they stand alone. It could all be a coincidence, but together is when you reach a parsimonious conclusion. It's the combination of the fact that we can't accelerate decay. It's the combination, with in turn with the decay thing, and that we have this beautiful gradient of skulls that appear based off of the previous premise to have existed at different times. It's the morphology, it's the genetics, and it's, to be kind of sassy, the fact that no one can answer that question for me. No creationist seems to be able to answer the question without kind of reverting to, I know you like this word, rescue devices. So that's kind of all I got to say. I mean, I'd love to have another conversation with regard to whatever genetic entropy or the age of the Earth, because as I said to Kent, a lot of it hinges on the fact that I believe the Earth is very old and you believe the Earth is very young. Because obviously, if it's old, the skulls make sense, and if it's young, then it makes sense for your worldview. But it makes sense based on what we kind of think. So I wasn't convinced, Standing. You didn't convince me, unfortunately. And I'm sure I didn't convince you either. So, but that's okay. I still had fun.
Having fun is the most important part. You're right. You didn't convince me, but you're right. It was a good back and forth. We could probably do this all day. I appreciate your responses. I appreciate your arguments and objections to my position. I guess going into, so if that was your conclusion, James, I just want to set my timer here. How long do I have? Just a few minutes, or I don't want to go over?
A few minutes. I'd say roughly the same as what Erica did. So I think it's
Okay.
like, what, 2-3 minutes?
I'll be really quick because I just wrote. So in my opening, I tried to address nearly everything she talked about in her opening. I just got a few things hanging that I didn't get to address. I'm definitely a stickler for that. So I'm going to address the couple things here. So DNA analyses anyways confirm that the supposed Homo species now all coexisted, intermingled, and interbred like we were discussing earlier. This is confirmation that they are all fully human. A few other things here that she discussed. Let me see here. Even at habilis, sediba, there's so much evidence now. Even some paleo experts are agreeing that these are classic examples of what happens when you piece together a so-called species based on a jumble of mixed bones. So what some evolutionists like Erica here are saying are your perfect transitional fossil from ape to man, it's actually a mixture of human and non-human bones. And they're simply an artificial species, a ghost taxa. That's why sediba, for example, Australopithecus sediba, is described as anatomical, with anatomical contradictions. For example, upper limb and shoulder anatomy, just like arboreal dwelling orangutans. And then you've got hands indistinguishable from modern humans. And this isn't the first time this has happened. This has all happened before. For example, one that comes to mind regarding the mixing of human and ape bones has occurred in Piltdown Man. So, which was just what they did. It was an ape jaw that was force fitted to a human skull. But unlike habilis and sediba, of course, I'm not saying habilis and sediba are a conspiracy by any means, but this was done intentionally, while habilis and sediba, not so much. For example, the evidence she brought in genetics, the similarity, I touched that. It's only 88%. Similar 400 million DNA differences exist between the two species. Pseudogenes have been overturned. Hierarchies, like we discussed, relative hierarchies, as I explained in the beginning, is just characteristics of designs. And then the Australopithecines, my last point. And then I think I covered everything. They share most of their features with living apes today. They're around 3 or 4 feet tall with long arms and short legs, ape hands and feet with long curved fingers and toes, opposable thumb-like toes, ape size and ape-shaped skulls, funnel-shaped rib cages with high-placed shoulders for suspensory behavior. And yes, they do have some uniqueness. I'll give Erica that in, say, morphology and anatomy, which is why the best conclusion is that these were apes and nothing more than just an extinct ape genus. And Erica probably knows the fact that a baboon bone was incorrectly assigned to Lucy for over 40 years. That doesn't really give me much confidence regarding the evolutionists and their many other conclusions on human origins. And I explained all the different species, Naledi, Neanderthal, Erectus, the hobbits, for example. These are all inbred subpopulations, explains all the degenerate features. Homo naledi, for example, explains the curved fingers. This is all expected after Babel, post-Babel, post-flood, isolation and inbreeding. Everybody disperses. A few unfortunate subpopulations migrate, remain isolated. We see this clearly with the hobbits.
You got 5 seconds.
Dwarfism, for example. So I believe I've made a strong case that human evolution is impossible and fits perfectly within a biblical model. Thanks so much, James.
Thanks
so much, Erica.
Thank you. Time. So very excited, folks. We will go into the Q&A. So thanks so much for all of your questions. It's been a wild one, really enjoyable. I really like having both of our guests on. And with that, thanks so much for your patience, folks. I know you're eager, so I am as well. First up, Brian Stevens. Thanks so much for your Patreon question. They said, does Standing for Truth think that Kent Hovind actually has 3 PhDs? Also, where is his, Kent Hovind's, CV? Or basically, for those of you new to a CV, it's like a academic resume.
I'm more concerned with the arguments one has. Actually, Tija made that point once, and I respected it. We're more interested in the arguments than, for one, ad hominems and looking at what people's so-called education may be, because some people with a lot of education may not have the best arguments, especially if they have nothing more than textbook answers. So I'm not really interested in responding directly to that. I'm just interested in the arguments that my opponent would have.
Gotcha. Thanks so much. Next up, we're going to try to speed through these folks. So if I'm going fast, forgive me. It's just because we want to squeeze as many in. Steven Steen, thanks for your super chat. He said, good to see Alpha Daddy, a.k.a. James, tonight. Gosh, you're sick. Nasty guy. You got to look out for him. Just, Next up, Michael Dresden, thanks for your super chat. He said, evolution is a myth, a fairy tale for grown-ups. A little trollish. Erica, if you want to respond, you can. You don't have to. It's up to you.
I won't give it the dignity.
Gotcha.
Okay.
Next up, Steven.
Good answer.
Michael Dresden. Oh, we got that one. Terry James, thanks for your super chat. They said, does different design mean different creator?
Does different design mean different creator? No, I mean, Honda, Volkswagen, for example, Chrysler, I mean, they have many different designs. You can look at the Volkswagen Jetta, which is a sedan. You can look at the Volkswagen Tiguan, or you can look at, for example, if you want to look at Chrysler, you can look at the Chrysler 300, the Chrysler minivan. I mean, there's different designs for the same creator. And of course, if God wanted to, for example, DNA, deoxyribonucleic acid is the universal code for all of life. And evolutionists say, this is proof that we're all related: pine trees, whales, banana plants, and apes, apparently. But of course, God is going to use a common code for his common design. If he used different codes, that would suggest a possible form of polytheism. Maybe there is multiple gods, like the Greeks or something. But one common genetic code makes the most sense given a biblical based God. So that's my answer.
Gotcha. Thanks so much. Jay Shy, thanks for your super chat. They said, standing for truth, genetic entropy is a lie by John Sanford. He misquoted Moto Kimura's, their study where Kimura left out beneficial mutations on purpose to focus on neutral mutations by genetic drift, not selective mutations.
I feel like I've had this debate so many times. That argument is a rabbit trail. There's never been any rescue device or argument that has debunked genomic degeneration. All serious population geneticists admit and agree that man is presently degenerating. That's why they come up with rescue mechanisms, synergistic epistasis, mutation count mechanism. When I debate these evolutionists, they use the most weak standard blog related arguments. I mean, at least bring forth things like synergistic epistasis and these mechanisms that these population geneticists are invoking. Let's discuss the data. No more of these ad hominems. John Sanford shows a larger zone of no selection than Kimura because Kimura's analysis of the problem was oversimplified. I debated someone a few days ago who's very familiar in the very exceptional in genetics, and we discussed this very issue for an hour and a half. So no, these new near neutral, low impact deleterious mutations, they are accumulating. But natural selection can only amplify the best beneficial mutations. It can only get rid of the worst detrimental mutations. Can't do anything against the near neutral mutations. They're accumulating in our genome, and beneficial mutations are no help. They are no help, and the vast majority of them are unselectable as well. So there's my answer.
You got it. Thank you very much. And thanks so much for your super chat, Terry James.
Thank you.
They said, if DNA similarity can be explained away by similar appearance, then why is the, correct me if I get this wrong, thylacine more genetically similar to the numbat than the wolf?
Is that question for me?
I feel like it is.
I think so.
It has to do with genetics and morphology. I thought it was for him. Is it for him or is it for me?
I thought it started off for you. So now, if you can ask it again, I'll answer.
I think it's for standing for truth. I'll read it.
Ask it again.
Yes.
If DNA similarity can be explained away by similar appearance, then why is the thylacine more genetically similar to the numbat than the wolf?
I think, as I explained earlier, we're going to have to, if you're looking at just mere similarity and anatomy, morphology, for example, we need to look on a DNA level. And we speak so little of the DNA language. I am going to look to, like I explained to Erica, DNA function. And this has been proven true over and over again. Endogenous retroviruses, ALUs, pseudogenes, they're all very important in our genome. I mean, they're important to sustain healthy life processes in the cell. And I can go on and on about the numerous functions that have been discovered based on our hypothesis. But that example, I would, if it's that similar genetically speaking, I would look to some form of function. I don't know if there's an answer yet to it, or if it's something that we need to discover in the future based on more genomic analyses and more genomic testing.
Gotcha. Thanks so much. Shannon Q says, Team Erica, in all caps. You've got a fan out there, Erica.
Shannon, I love ya.
Brian Stevens, thanks for your super chat. He said, Standing for Truth's sources for footprints is, quote unquote, the conversation.
No, I've got plenty of sources. Actually, too many sources on that one. Like I said, I looked heavily into that footprint. I'm looking forward to the source that Erica explained and is going to provide to me, because what I've found in the source data in the primary literature, they all agree that these footprints are modern, anatomically modern looking human footprints, and they're trying to explain it away with some form of possible extinct gorilla, or I mean, there's a lot of, I guess, data out there. So like I said, I'm looking forward to Erica's sources, but I've got a ton of sources. I can certainly provide them to anybody who is interested.
Gotcha. Thanks so much. Next up, Stephen Steen, thanks for your super chat. He said, my wisdom teeth fit in my jaw. Am I the missing link?
Hey, I had all four of my wisdom teeth too. Wisdom teeth are an interesting little facet because they're kind of on their way out, but not quite. Some people have no problem with them at all. I had problems with mine, and that was a wild after surgery experience. So I'll put it this way. You are the missing link. There's no other explanation.
Gotcha. Thanks so much. Next up, appreciate your question. Brian Stevens says, why do you ask Erica to use scientific... So this is for Standing for Truth. Why do you ask Erica to use scientific sources to disprove your blog from, quote-unquote, the conversation by a, quote-unquote, political analyst?
There's plenty of sources. I actually have innumerable quotes from all types of paleoanthropologists that agree with what I've been saying. I provided a paper in my opening regarding my hypothesis that these so-called human ancestors are nothing more than inbred subpopulations that experience rapid genetic entropy due to the accumulation of mutations. And we've got an evolutionist here who's written a paper recently pretty well confirming the exact same thing. Of course, he still holds to evolution, and he admits that there's something odd with the data, but I've got a ton of things that support me, quotes, sources. So if Brian Stevens is serious about it, I'll provide those to him after the debate.
Gotcha. Thanks so much. Next up, Terry James, thanks for your super chat. They said, Standing for Truth complains that Aron interrupts, but it's okay for him to interrupt. How do you like them apples?
You know what? I think this was a good debate because Erica and I both, I think, like to talk a lot and talk about a lot of different subjects. But anytime there is interruption going on, James, being the best moderator on YouTube, stopped it right away. And thanks for that, James. So if Aron Ra and I get in on this, hopefully we're not just talking over each other the whole time, because James is going to make sure that doesn't happen. So
that's nice.
Is that a planned debate? I'm interested.
Yes, it is. In fact, this is an excuse for me to show the thumbnail again, because I'm excited. This is going to be an epic one. So basically, Aron Ra and Dr. Mick Leroy, I mispronounced it earlier, sorry about that. Dr. Mick Leroy and Aron Ra will be debating on creation versus evolution, which best explains the evidence. And that's coming up in Dallas. It'll be in person. So we've actually... Team Skeptic.
That is so exciting.
It is. Team Skeptic, I have to give a big thanks. So if anybody out there, if you know Team Skeptic, give him a big thanks, because he was so kind. He's got a channel called Team Skeptic Debunks, or I should say, to be neutral. You can say debunks arguments without necessarily meaning that a view is true. He challenges the flat Earth position, and he's a very good debater. And he's also very kind because he helped us get the venue for this debate with Aron Ra and Dr. Mick Leroy. So that's honestly going to be a blast. I'm pumped for it. But yes. Erica, do you live in Texas? You have to go.
I don't. I know. I think being in the UK for most of the year means I'm a little bit out of that range. But I would love to come. Hey James, come to the UK. Let's do a debate there.
Let's do a debate tour in the UK.
I'm actually, no joke. There's I was thinking about it earlier today of whether or not they could actually happen. And it's not.
Oh, that would be so cool.
It's not impossible. So it's definitely a possibility.
Hey, if you do that, I'll take you out and get you a drink at one of my favorite pubs.
That's nice of you. I appreciate that. And someone just asked in the audience, are there tickets? We don't have tickets. It's going to be a free first come first serve. It's actually going to be in a tattoo parlor. So we're kind of doing almost like the Bible and Beer Consortium. They go to pubs. We will do it in this tattoo parlor, and it's going to be a lot of fun. So I'll let you know the exact address as we get closer. And but yeah, so hopefully we can fit everybody in there because I think there's going to be a lot of people. So it'll be a lot of fun. And next up, thanks for your super chat, Athena, goddess of wisdom and warfare. They said, standing for truth is an all right guy. He's just wrong. What do you think of that, standing for truth? You like that?
There were some really, really good play on words regarding SFT. So, whoever can give the best play on words for SFT, I think what was one of them? Standing for lies is a good one, but it's $25. No, I'm just kidding. But yeah, Athena, good guy. I've done a lot of discussions on his channel. I got nothing bad to say about him. I would say he's a nice guy, and he's wrong as well.
Gotcha, thanks for that. And so sorry about the sound. I played the boom button because when I read that super chat, I was like, oh, good jab. And I played the boom button, and it was the most frightening, terrifying scream ever, and everybody's like, dear gosh, what was that? So thanks for your patience, guys. All right, stupid whore energy, as she likes to be called, thanks for your super chat. She says, standing for truth, how do you explain the small population of Sumatran orangutans exhibiting high genetic diversity but thriving?
To be honest with you, I'd have to see the source and the paper on that one, but I'd bet some money that there's a pretty decent explanation for it because it sounds like he's probably grasping at straws. But yeah, if he wants to send that, I'll give an answer afterwards. Never heard of it.
Gotcha. Appreciate that. And thanks so much for your super chat, translator Carmium. Sorry, that's embarrassing. Okay, they say, standing for truth, how can you complain about a bottleneck of a few thousand if you believe in the post-flood Noatian bottleneck, which was much smaller still?
Good question. Good question. That's usually the question I get after I bring up the fact that their so-called population bottleneck is not even remotely feasible. But ours is very easy to explain. I mean, based on our model, okay, I'll take it slow for them, use really small words. We start with just two people, okay, constituting a very extreme yet benign population bottleneck, okay? Then, and you can just read this in the Bible, 10 generations later, a second single generation bottleneck of just what? Whoever knows their Bible, just 8 people occurred at the time of Noah. Both bottlenecks were very brief, just one generation, and then followed by explosive growth. These cases, there would be almost no previously accumulated mutations, especially if we explain the vast majority of DNA differences as functional created DNA differences, therefore no inbreeding effects. That's why the limited human genetic diversity we see is good for us, but a huge problem for evolutionary theory. And as you can see from the evolutionist explanations, it leads to unrestrained storytelling. So that's my answer.
Gotcha. Thank you very much. Next up, trying to move through fast, Stupid Whore Energy, thanks for your super chat. She says, if mutations are degradative, why do viruses often evolve complex structures like HIV has evolved complex gated ion channels?
So he is he talking about virus? So the HIV one, is he also referring to like endogenous retroviruses, like viruses in general?
I'm not sure if she only mentioned HIV.
You know what? I'd have to see, because I know the explanation for the function found in endogenous retroviruses is that these functions are co-opted, just like pseudogenes, for example, they're co-opted. But the thing is, that's just theoretical, right? We didn't actually observe these functions being co-opted, so I'd have to see if that's the same thing going on with the what's going on with the HIV as well.
Gotcha. Thank you very much. We appreciate that. And next up, Stupid Or Energy again, coming at you. She says it's been
He's busy.
That's right. She says it's been confirmed that that telomeres in nearby DNA undergo lots of mutations. So of course you'd expect this kind of DNA to be divergent and degenerate.
So she's talking about the telomeres. I guess she's admitting that the sequences that we should be seeing, if there was a chromosomal 2 fusion, right, we should see these remnant telomeres, and there's a struggle to find these because they're degenerate. So I guess she's admitting that they're degenerate, at least, which is kind of helps my case. But I would just ask, why were the telomeres even involved in the fusion to begin with? Their job is to prevent the fusion. So as far as I know, the telomeres don't occur in the middle of genes. Genes are not around telomeres. So I mean, there's so many discrepancies to the argument, to the story of the chromosomal 2 fusion. So
Gotcha. Thank you very much. And then Erica, if you'd like to respond, I know Standin for Truth gets a lot of these, so if you want to like add something on some of these, like feel free.
I mean, I would just say on that we do have the remnants, and they're in Tompkins' own paper. So that's why I kind of like sassy pointed up at this. I was like, hey, there they are right there. That's all I have to say.
Gotcha. And we do have a question.
Actually,
Hold on.
If you have a question for Erica, perfect, because I got to run upstairs real quick.
We do have one.
Taking a 1 minute break.
Gotcha. Thank you very much. And so, Mitchell, thanks for your super chat. They said, question for Erica, if creation and evolution both include nested hierarchies, what mechanism naturally created your quote-unquote magic cell via abiogenesis?
Well, I think that's really a great question. I mean, basically the field of abiogenesis is booming. I think it's kind of interesting that they asked that in a human evolution kind of chat, given that abiogenesis is entirely unique. But it all starts with chemicals, right? I mean, chemical reactions, they appear to have no real rhyme or reason to them. But as far as I know, it was amino acids that kind of began to clump together and sort of form these proto strands of like RNA at some point, if you subscribe to the RNA world. That's on my field. I wish I could give you a better answer. Ask Jackson Wheat. He knows this stuff.
You got it. Thank you very much. And I assume that Standing for Truth is still occupied, so I'm going to just quick mention a couple of things. First, stoked to have you here, folks. We rarely talk about the philosophy of the channel, but I think it's worth mentioning that we are basically trying to provide a non-partisan platform, so we don't take any stances on the issues. We don't have after shows where we say who won or lost or anything like that. We are trying to be fully neutral, and despite being imperfect, that's our goal, and we want you to feel welcome, whether you be Christian, atheist, Muslim, one of the many strange creatures in between all these different worldviews. We're glad you're here, and it sounds like, Standing, are you back?
I am back, my friend.
He's
all
back.
That tea and water will get to you after a couple hours.
Nasty. Thank you. So, very good. I appreciate that, and we do have a question.
No problem.
Don't worry. Stupid whore energy. Oh, nasty. So
she's putting your kids through college there, James, with all those super chats.
She does do a lot, and she says, how much for James's snot-filled rag I will pay. Nasty woman. Very just disgraceful.
A lot of nastiness going on.
You've got some good questions.
Unfortunately, Steven Steen already offered $20. That's hard to beat. I mean, he's a nasty guy.
You're missing the big money here, my dude. You got to start capitalizing on these apps.
The snot rag. That's funny. Oh boy. Subtracted. Thanks for your super chat. They said, "Hey, Standing for Truth, why is the FOXP2 gene expressed in the chimpanzee genome as a mutation?"
What I know about the P2 gene it's the gene associated with language, but it does far more things than just that. So the fact that it's broken doesn't, give evidence that if it worked, they would. I'm not sure if I'm answering the question properly. Can you?
Yes, let me pull it up 1 second. And I've got to undo this. Hey, Standin for Truth, why is the FOXP2 gene expressed in the chimpanzee genome as a mutation?
Okay, so it as a mutation. So if it was originally functional, like I said, it's associated with language, and if it does far more things than that, and now it's expressed as a mutation, I mean that would just be evidence of genetic entropy after the fall. I mean, some functional genes can be the result of mutation if that occurs.
Gotcha.
Okay, if I jump in real fast on that one, James, just
You
like
bet.
a little quick anecdote. Since Standin's getting all the questions, why is no one asking me any questions? I'm bitter about it. But I think that's an interesting question. I'm not positive that the FOXP2 gene is language because I don't really know that gene particularly well. But if it is, the interesting thing is that chimpanzees have some like 86 or so unique gestures that they can combine to get across messages, and human infants display 46 of those between the ages of 1 and 2. So they do indeed have a sort of proto-language, and that can be extended to geladas, whose chittering follows Menzerath's law, or vervet monkeys, who have their own vocabulary for predators. So it is very interesting that we actually see a gradient of language, very proto-language, in living primates.
Gotcha.
Yeah, I think because just real quick,
Super
quick. I promise.
Okay.
Just like I said at the beginning, I just wanted to make sure I was answering it right.
Go on.
It can be associated with more things, more functions. For example, and then I'll compare that to protein moonlighting, which back in the 40s, one gene, one protein, one function was the thinking. Now we know of protein moonlighting. These proteins are doing unimaginable things. It's like a multifunctional tool. So some of these genes have more than just one function. So it being a broken gene or a mutation is not, I don't think is going to convince me of ape human evolution.
Gotcha. Thank you very much. Next up, got one from Subtracted. Thanks for your super chat. He said, hey, Standin. Oh, we got that one. That's embarrassing. Okay. Stupid Horror Energy. Thanks for your super chat. She says, are you aware that two proteins don't need to have the same amino acid sequence to perform the same function?
Is she referring to third position redundancy?
Let's see here. I'm not exactly sure.
Say it again.
Yeah, and you know what? A lot of those degenerate codons, for example, are also now, there's massive literature suggesting function. Because you can look to somebody like Dr. Herman Mays, who also looks at DNA level hierarchies, and he'll look at neutral variation, third position, degenerate codons. But now, even those are proving to be functional. They work to slow and speed processes in the cell. So it all falls in line with pre-existing heterozygosity. So yes, I am aware of that.
She followed that up with a quick one where she said, gene duplication is a very old concept. And she also said, I love Nickelback. I don't know why I made that last part up.
I mean, the evolutionists will often look to gene duplication as a way to add novel information to the genome. But I mean, if I give you a copy of a book that you already own, and then I start introducing typographical errors into the second copy of the book you already own, now you just have a more messed up copy of the original book. So it's just more evidence for genetic degeneration. It comes down to net gain versus net loss. So that ain't going to help.
Gotcha.
Quick add on that, though. What about natural selection? Ooh. That's
right. Natural selection, though, the problem is it can't act upon those near neutral mutations that are unselectable. Those are the ones that build up. Sure, it's going to amplify the best beneficials and get rid of the worst detrimentals. It's the near neutral mutations that accumulate and degenerate our information system. So that'll have to be the problem you'll have to address, Erica.
We should have a conversation about genetic entropy. I've heard you speak to like CRISPR and stuff like that before, and I would be very interested to have that chat. I'm not very well versed, as I've said, for like the 20th time. I'm not very well versed in genetics, but still, it would be an interesting chat.
Yeah, I think that'd be a blast. You've been fun to talk to. We can definitely set that up.
Sure. Sorry, James. We started talking again.
Oh, James, you still there?
No problemo. So I appreciate that. And I think that's a great idea. You guys ought to. And the next one comes from Stupid Horror Energy again. She's persistent. She says, how do you explain the fact that ERVs, or dead viruses, are in the same exact places in humans and chimps, but not other species?
So good question. Regarding ERVs, it comes down to the fact that are they really the result of ancient viruses? Because now we know that the evidence suggests that these endogenous retroviruses and these retrotransposons, for example, they're indeed functional, and not just functional, not just low levels of function, but these endogenous retroviruses, they have crucial functions. They help regulate genes and even determine cell types. So like I said earlier, the evolutionist now has to claim that they were co-opted, but that is just more fancy storytelling. So these are important DNA elements, just as we'd predict based on created genetic diversity.
Gotcha. Thanks so much. Next up, we got one from Cy Gart. Thanks, Dr. Gart, for your question and super chat. He says, inbreeding requires a much smaller population than 2,000. Chromosomes are different between species because most of the genes on Y are not essential.
Yeah, I don't really think that matters because if they're different or not, I mean, the fact is, if we're talking about human evolution, chimps and humans do have a Y chromosome that's very, very dissimilar. And if you want to look at the Y chromosome, well, they can be traced back, proving one paternal line. And some of these degenerate chromosomes regarding the Y chromosome is just more proof of genetic entropy, the crumbling genome. That includes the Y chromosome as well.
Gotcha. Let's see.
Hey, I'm very flattered that Cy watched this conversation, though. Cy, you're awesome. Love your stuff.
I like Cy. I hope you enjoyed it, Cy.
Yeah.
Gotcha. Thanks so much. And by the way, so we are trying to, I'll mention that 99% of you all here are honestly so terrific, friendly, and you like to debate, but you're not like, like being like abusive, which is great. You're just superb.
Terrific.
And there's maybe like 1% that sometimes is a little bit like more fighty and a little bit more aggressive. And so in that case, just want to ask if you would be so kind. We're trying to like change the ever-changing culture, trying to mold it into like, yeah, we're going to try to only ban hate speech. But we do want to have the mods hopefully just say like, hey, like if somebody's being abusive, like, hey, can you do us a favor and not call somebody an idiot? Something like that. So we're still going to try to like not regulate people. We don't want to like control people. But at the same time, we want to like kind of naturally, organically, like through your own free will, foster that kind of spirit of like kind of like warmth or friendliness. You know what I mean? Does that make sense? So
James, that's so diplomatic. That was such a diplomatic way of saying, cut that out now.
That's funny. My hope is that hopefully it'll just be... And like I said, 99% of the people, very kind, like it never are abusive. Once in a while, though, it's kind of like, kind of. But
yeah.
So with that, next up, we're going to try to fly through as many. We just got through the super chats. So thanks so much for those, folks. And we are going to jump into the other questions. I will try to do this as fast as possible. We're not going to get through all of them. I'm really sorry about that, everybody. There's a lot. And I want to respect the debaters' time. We've already...
I just have to jump in, James. To be honest with you, I've only got about 2 more minutes. So if I'm guessing most of the questions are for me, so you can go through him as much as possible. But
yeah, I know what you mean. I know what you mean. We've gone over your time, and I'm sorry about that, you guys.
I'm sorry.
No, it's been a blast.
Don't worry about it.
And
all
so,
good.
Love your channel, James. You put on the best debates.
We usually try to do like an hour and a half, 2 hours tonight, but didn't quite do that tonight.
I lied.
So
hopefully it was one to remember.
Yeah, absolutely.
Honestly, it really was. People, honestly, there's just a lot of positive feedback. People have really been enjoying this and engaged, and so I've definitely enjoyed it. And we'll try to fly through just maybe 1 or 2 of these. Actually, what we'll do is maybe like 1 or 2 for Standing, and then we'll do 1 or 2 for Erica, because that way Standing you can go. And so 1 for Standing, we will do this one from Rayward. This came in early. They said, I already know Standing for Truth will evade. Can you describe exactly what a transitional form between apes and humans would look like if you were to be convinced evolution was true?
Yeah, no, I'm not the one. If you'll actually hear me rarely even bring up transitional forms, because when it comes to transitional forms, I'm not difficult with it. I think a transitional form would be the blending of two separate creatures. I mean, if you look at something like maybe Australopithecus sediba or habilis, or even the Piltdown man back in the day, you know, these so-called human ape ancestors that are clear evidence of the mixing of Homo and ape bones. I mean, those would be the probably close to the perfect transition. But as I've explained many times, when it comes to transitional forms, our model too predicts the mosaic, the transitional form. For example, I point to products of human design that seem to span two categories, just like you can find certain animals. Remember, the majority proves the rule. They can point to some, say, reptile mammal transitions, or they can point to tiktaalik, for example. It's going to need to be the rule, these so-called mosaics. But an amphibious assault vehicle, to the evolutionist, would be the perfect transitional form. So it would have to be a blending or a combination of two categories, right? A really good combination of human features and a really good combination of ape features.
Gotcha.
James, would it be okay if I said something super quick?
Yes.
Just like mega quick?
Correct.
I would compare that to being like, okay, so we have Archaeopteryx, and then the human, comparison to that would be an airplane with wings that doesn't fly and rolls around like a car exclusively, which we don't see. So, transitional forms are kind of indicative of kind of rigging existing structures. It would be all I would say about that.
Gotcha. Let's see. We will, standing for truth, we can cut you loose. We know you got to go, so we appreciate you being here, and thanks again for your patience. I know we went over the time that we promised.
I was wrong. I'm
Sorry,
sorry.
it was a blast.
It was all a
really
mistake.
enjoyed this debate. Good questions from the audience too.
Yeah, absolutely. Thanks for chatting with me, Standing. I had a great time.
And
Thanks so much, Erica. Hopefully we can do it again soon.
Yeah, absolutely.
It was a lot of fun.
Sure.
Quick.
We'll email. We can talk about it when I send you all those Crete papers.
Sounds good. I'm looking forward to it. Thanks.
All right. Great.
Thanks for doing it, Erica, and James, thanks for setting it up.
thanks, Standing. Take care.
Bye.
Goodbye.
Okay, we'll do one more question. This one would be for Erica. And so sorry, folks. I know that we can't get through all of the questions. Is that you, Standing? Can I hear you lurking?
I'm still here.
Okay.
I want to wait till the very end, but that's not fair. Erica's had all the questions. Can't I get some?
Yeah.
That's funny.
Yeah, really, I've been overloaded. It's a good problem to have, James. Too many questions. It's the best kind of problem.
Sure.
That's good. It is. And so let's see. Landon, thanks for your question. He says, Erica, some microorganisms display decision-making abilities and can be classically conditioned. Would you be open to considering such intelligence as evidence for design?
Well, I think it's very difficult to kind of draw that line between what we would consider, like a cognitive prowess that we see in like corvids and cetaceans and like anthropoid primates, and what we would see as just a response and stimulus. So I'd be interested in seeing the papers for myself because I've never heard of that. It sounds really cool. I'd be interested in seeing the papers and seeing if perhaps they kind of managed in their methodology to somehow rule out that this is just sort of a chemical just response to a stimulus.
Gotcha. Thanks so much. And with that, want to say thanks so much, folks. It's been a true pleasure. A lot of fun tonight. Both of our speakers are linked in the description. So if you liked what you heard, and you're like, yeah, me likey, then I highly encourage you. You can always check out their content or their links in the description.
I like that.
So very fun. It's been a great debate. And also, we are very excited. If you want to, don't feel obligated, but you've already heard, if you're here, you're probably a creation evolution type of person interested in those topics to some extent, right? So my request is, if you'd like to help the channel, you don't have to. Don't feel obligated. Only do it if you'd be like, I'd like that. I'm going to put the Aron Ra link in the description right now. If you have friends who like this topic, or if you have a group or something that you want to share it in, we're trying to get the word out about this. It's exciting. We are glad that Aron is willing to participate. In fact, actually, yeah, what I'll do is I'll put it in both the live chat and then I'll also put it into the description. Is if you want to spread the word about that, that helps a lot for the channel to grow, just because we are trying to, again, basically build this platform where everybody's welcome, whether you be, like I said, Christian, atheist, gay, straight, no matter where you're from, Republican, Democrat, we're glad you're here. And that link is in the live chat right now, and I'm also going to put it in the description. So with that, though, thanks so much, folks. Share this debate as well, as this has been one of our, honestly, I've had so many, it's been like 200 debates we've had, and it's like the support for this one was like through the roof, even before it began. This one had more likes than the Aron Ra debate. I'm not joking. So you guys,
I don't believe that.
We'll take that as a compliment.
I'm totally serious. So it's
that means we got to do it again, Standin. We have to come back and talk about
Yes.
genetic entropy and the age of the Earth. We have to. There's no option.
It's a must. Round 2's coming.
Yeah, it's got to happen.
We'll make a good trailer for it. Pump people up.
There you go. There you go.
You betcha. So thanks, folks. It's honestly a pleasure. This always just boosts my mood. I'm always like stoked to be here every time. So it's been a pleasure. We hope you keep sifting out the reasonable from the unreasonable. And thanks so much to our guests. We appreciate you guys being here.
We're glad to be here.
Pleasure.
Have a good time.
Absolutely.
Great.
So with that, take care, folks. It's been a pleasure.
Remember, the Force will be with you, always.